Good morning, everybody. My name is Ed Bilski. I'm the Associate Provost for Research and Scholarship at the University of New England, and I'm going to get to introduce many of the speakers today. This is a great pleasure. This is the second year in a row that we've done this Capstone Symposium, and it combines both the undergraduate science and research scholarship that's taking place at the University of New England, along with a special program that combines science, medicine, and entrepreneurship. And I'm going to try to, in the next 5-10 minutes, put this into a perspective of what the University of New England has been doing to help stimulate economic development and growth in the state of Maine and in the region by partnership, and also tie that back into our research and scholarship missions and our academic mission of educating the next wave of healthcare professionals and other career opportunities for our students and our graduates. To start, I want to thank a few key people that made this all possible. The sponsors, who many of whom have displays out in the atrium in the third floor here, including Clear H2O, which Jay Palmer is here, Marine Plastics, I think Sam Tarbox is here, the Goulet Foundation, Brad Goulet is here representing the family, I think Danielle is also here, and many others are listed in the sponsorships. In addition, we've got some people that behind the scenes put a lot of time and effort planning this summer. Jordan Falloon, who is, not sure where Jordan is right now. I think she's probably getting something to help with the program. But she's going to be seated down here. She took control of this because I just was not going to be able to do it all myself this summer, and she stepped up with that. Denise Giavellis in my laboratory has spent inordinate amounts of time making sure everything goes smoothly. Henry Furland. Henry, where's Henry? Everybody's working still. There's Jordan. Henry, are you here? Henry's quiet and unassuming. He really also stepped up. He's going to be a third-year medical student. And have I forgotten anybody? Along with the interns who have put a lot of time and effort into this as well. Okay, so to put this all into context, this is a pretty young institution. It came together in 1978 with St. Francis College and the brand new New England College of Osteopathic Medicine. And they had two things going for them in 1978. One, they decided to come together and call themselves the University of New England, which immediately gave itself a regional presence, which was going to be very helpful in marketing the institution. And some thought it was grandiose, and the first president, Jack Ketchum, said in a typical Mainer fashion, we're going to grow into that name. And I think if you look over the last 30 or so years, we have grown into it, and we're going to continue to expand our presence in the New England region. In addition, we had a world-class location, which you're privy to today on a gorgeous day in the end of July. And that also was certainly a recruitment tool. I use it as a mooring for my boat. That was one of the main reasons I came here. Actually, there was many reasons I came here, and besides the location, I saw the potential about 11 years ago. We talk about entrepreneurship, and sometimes we think about that in the context of big Fortune 500 companies, maybe a startup biotech company. We don't so much think about entrepreneurship for a nonprofit or a university or college that's doing higher education. But in fact, entrepreneurship can be defined in different ways. I like this definition. It basically says we realize that there's needs. We put teams together to fully exploit our resources and things that we're passionate about to create something that is meaningfully unique and to move that mission forward and to accomplish great things. And when we talk about our inherent DNA at the University of New England, I think one of the really important ones is student-focused research and scholarship. That the faculty, from the beginning, the inception of this university, knew the value of it and invested heavily in it, both time, effort, and other resources that were available. And that has helped fuel our successes over the last 30-so years. In addition, we have centers of excellence that have tried to focus resources on things that we can be very good at or maybe outstanding at, and including things like marine sciences, given our location, some of the investments we've made. Neuroscience is one of the other ones that has certainly had a success story, and I'll give you some examples of that. It started relatively quietly. We had some founding faculty that had to deliver neuroscience curriculum to the medical students, and we're fortunate to still have Barbara Winterson and Frank Willard, founding faculty members in 1978 and 79, being still part of this ongoing efforts. And another person who was a key individual, Peter Morgaine, who passed away about 2 years ago, had a real impact on this university. He came from world-class research institutions, had a place in Kennebunk, Maine, and found an attachment with this university and helped grow it. We had some other faculty, including Dave Mokler, who's here in attendance today, and a few others who helped with the undergraduate curriculum as well. We had a pivot point, I think, about 12, 13 years ago. We had a dean named Steve Shannon at the time who wanted to grow biomedical research in the college, and we had a brand new lady that he recruited, Amy Davidoff, who's still on our faculty here. And together, they created some innovative policies that allowed us to recruit scientists that could kind of stimulate the next wave of expansion of the research programs. And I loved Amy's mantra, and it rings true today. She wants to have a better deal for whoever comes after her. And that's the way you advance programs, with selflessness and an eye on an overall mission vision. So this is what we are today. This is an amazing group for the size of a university that we are. This is the Neuroscience group, the Center of Excellence in Neuroscience. And all told, through both direct and indirect affiliations, we've got something like 40, 50 faculty members and researchers. We have a mission, and that harnesses our passion for understanding the complexities of the nervous system and applying that knowledge to improve human quality of life. And I think that's something that we're still kind of growing into as we think about clinical translational research opportunities, public health and neurological disease, prevention, and working with groups like the Goulet Foundation to accomplish that. One of the things we've worked very hard at is to be competitive for major NIH, National Institutes of Health, awards. And one of those is the COBRE grants. These are Centers of Biomedical Research Excellence. Ian Meng is the principal investigator on a $10 million award that we should publicly announce within the next week. We've been delayed with NIH and the summer slowdown in Washington, D.C., but this will be transformative for the University of New England. It's going to invest in many of our best junior faculty to give them the resources they need to become independent scientists. And in addition, it's going to be offering core services that many of our faculty and students are going to be able to take advantage of. On the economic development side, the state of Maine has had some seminal reports over the last 5 or so years that have looked at where we are as a state and where we want to be. And biotechnology has been one of the emerging clusters that they've felt needs to be invested in and it can grow. And they've been also very critical, though, of the nonprofit institutions that have received state funding to help build that infrastructure. While we've had some great successes as nonprofits in building the basic science component, the results with the translation and the development side of research and development have been lackluster. And so that has caught the attention of the legislators and the taxpayers. They're investing their money, and we have a mission not only for the education of our students, but if we're going to take state money to help advance economic development for the state and grow the workforce. And so one of the things that they had recognized in 2008 was that the investments that they were making, there was not enough collaboration with industry. And so it was kind of nice. We were the only company back here in 2008 that had a contract with a Maine-based company. It was one of our spin-off companies that was founded in 2007. But we took this to heart, and we started to figure out what was important, critically important to a small company in Maine, or maybe a mid-sized company in Maine, and how we could work with them. And so that was in part flexibility, listening, and because we were a private, relatively young university, we had the resources and the abilities to work much more quickly than, say, a larger system that's more entrenched with the way that business as usual. So I think that definitely gave us an advantage. And just some examples that will be highlighted today with some of the summer interns. Clear H2O is a Portland-based company. They make gel hydration products. They do have some taste samples out there. Jay Palmer brought some. They're good for animals and they're good for humans. And they started working with us on both the College of Osteopathic Medicine and the College of Pharmacy with our analytical core, and we're able to help advance their product lines by doing fundamental research to see if their products work, if they distinguish themselves from the competition. And so this is a very quick path. We can do turnarounds, as you'll see in the presentations, to give them feedback that they can then modify their products and get them to market. The add-on products I thought was interesting because they had a need to elevate one of their holders for their cups. And we had this other Maine-based company in Biddeford, Marine Plastics, which you'll hear a presentation on, that was making custom chambers for us. And we think we can open up new business opportunities for this other company by marketing to neuroscientists and other life sciences people well outside of the realm of marine science, where they've got their name and their brand out. So in terms of quality and benchmarks, one of which, the easy one, is do you look at extramural funding? So the university attracts contracts, grants, and other types of investments from a variety of different sources. And if you look over the last 5 or 6 years, there's been a steady increase in that extramural funding. People are investing in us. They think that we are a quality product and that our faculty are competitive for these grants. And nowadays, funding for the NIH is down well below 10%. So we're competing with the very big universities and colleges, and we're doing it successfully, and we're on an upward trajectory. We're also seeing a lot more publications. We're seeing more people competing or putting in submissions for these grants. So all indicators suggest that we are doing something fantastic on that benchmark. I talked about meaningfully unique. What does that mean? Why are we doing all this? One is to be true to our mission of educating our undergraduate students, our professional students. Also, we do a lot of community outreach, and I think that is meaningful. We are working with the school systems. Under the leadership of Mike Berman in the psychology department, we came in contact with over 1,500 students this past academic year. That was up from maybe 300 the year before. And all indicators are we're involving more and more people to do this. We're starting to get some momentum. We're going to get some external funding to support this. This is important, though, because it's raising brain awareness. It's working with people like the Goulet Foundation on head injury prevention, awareness of brain injury, things like that we can bring to the school systems and deliver on that. And I'll end, before I introduce our first speaker, with Dan Carr, who's our keynote speaker. I love this quote. It ties into a quote that he gave with a keynote address for a graduation. I think it was a public health presentation. But he challenged the graduates, and I think this is a challenge that I would make to everyone here at the University of New England. I'm going to urge you to overreach, get involved in projects that are larger than yourself, to adopt a youthful and optimistic and even unrealistic perspective that underestimates the work-to-reward ratio. Basically, that means that we're going to go in maybe a little bit naive, a little bit over-enthusiastic, but it's amazing when you bring a group of people together, you can accomplish things that you never thought would be possible. There's certainly a lot of hard work involved with that. I think one example of this is the program that we do for the summer researchers. So with that, I'm going to turn over the stage to Alex Colville. He's our first summer intern. Alex is a chemical engineering major at Northeastern. I think you're from Scarborough. Got that right. And he kind of had a project that's a hybrid. He did conduct some original research, and in addition, he was working with Clear H2O, kind of a development of a business plan. So without further ado, Alex Colville.
Thank you very much. Welcome, everybody. So yeah, as Dr. Bilski said so wonderfully, I'm Alex, and I worked with Clear H2O. And what we're trying to do is kind of change the way medication was delivered to lab animals, which was kind of interesting. So, the history of Clear H2O. Many of you may know Charles River Laboratories down in Boston, a huge drug development company, and they had a problem. They were shipping lab animals all over the world, all over the country, and the water bottles in the trucks were shaking. The water was getting everywhere, and there was no water left. The animals weren't able to drink, and dehydration and health problems led to huge headaches for Charles River Laboratories. So they realized that they needed to make a quick change. So they actually went out and looked for companies that could produce a hydration gel for the shipment of these lab animals. And they actually contacted like 15 or so companies, many of which weren't really interested in branching out into kind of a new field for them. But luckily, they came across Schlotterbeck and Foss right here in Portland. And that was a real breakthrough because they were thrilled. These were leaders in gel technology and had been in the food industry for a while with food gels, and were very happy to make the transition. And eventually, the owner of Schlotterbeck and Foss asked for permission to patent the technology themselves. CRL really wasn't interested in producing gels. They're interested in drugs. So they were thrilled. And Paul Lioli, the owner of Clear H2O, said, let's go. So today, located on Preble Street, and they produce a variety of products to nourish and enrich these lab animals. And as you can see there, it's quite a variety because you have your standard lab animals like mice and rats, but also primates and chicks. And as you can imagine, raising chicks is extremely different from raising mice in like an ultra-clean environment, like a vivarium. So that leads to some interesting problem solving for the sales department at Clear H2O, which I thought was really cool. Another thing about the company is they utilize a lot of distributors around the world. Belgium, Germany, Austria, pretty much anywhere I name, they'll have a distributor there. So that allows them to really be global in their market. So products. The original hydrogel, which as I talked about was the water problem, they've expanded to a variety of products, such as the diet gel, which are aimed at kind of mimicking normal diet and NIH standard diets, as well as a different sort of diet gel, the Boost, Recovery, and Criticare, which are very useful for post-op. Post-surgery, animals, just like humans, will lose a lot of weight. And researchers really don't like this. You want homeostasis. Everything in research needs to be the same. You don't want huge fluctuations. That will mess with your data. So that's where these products come in. And they're really helpful for that, maintaining homeostasis. As was mentioned earlier, the day I actually first arrived at Clear H2O, they made me try all of these things. I wasn't very thrilled about it, but I accepted my role. I kind of questioned if I was the guinea pig or the one doing the experiments, but I found out. You guys can try them later. They're actually pretty good. And one that really mimics that is the Fiber Bites. They're actually gummies. They look just like what you find on like kids stores and grocery stores. And they're for primates for dietary tract, so they're probiotics. And finally is the Metagels. What's important to note about these two, though, is another huge focus is the backup system. So labs need backups because bad things can always happen, disasters. Like water mains can break and PIs can leave for Bermuda on sabbatical. So you always have to worry about funding. So the Metagels. You know, it's really bizarre. It turns out mice don't actually like pills. You would suspect it with humans. We love pills, but mice, not the same. So this is an interesting problem. Currently, the solution is there's oral injections with syringes down the mouth. If you miss, though, and you go down the wrong pipe, seizures can happen. And those are no fun for anyone. Terrible for the animal. And the other solution, which is pretty good, is injections. But those can be pretty time-consuming, and they're not the most cost-effective thing. So we're working on a different approach, and that's where the picture on the right comes in. If you give a mouse a cookie or a gamma-irradiated gel of hydrocolloids and sucralose, it turns out they want another. So this is where I see the company going, and this is with the Metagel products. Currently, they're working on antibiotics. As you see there, that's the FBZ. It's actually a pinworm treatment. So if you have an outbreak of pinworm in your mice, all you have to do is pop open a couple of lids, throw them in the container, and they're cured, which is very hands-off and very easy instead of having to do all these injections, treatments, and measuring, which is great. And the sucralose is actually a generalized Metagel, which means that whatever medication you have, you can actually pop it in and use it that way. So where they're looking to go is analgesics, and that's where we come in the pain department here at the CEN. And so they're really looking to grow and looking for funding for these studies. So I'm actually working on right now a Maine Technology Institute grant, a seed grant, in order to allow this expansion and this new flow of money, because they need money for studies for things like efficacy in mice and shelf life and stability. And that's all really important to developing these analgesics that'll be used in post-op care in mice, because just like humans, mice feel pain too, and ethically, it's our duty to make sure that they're not in pain. Okay, so how does Clear H2O make itself stand out in such a variety of biotech companies? One of the huge things is that they only really care about gel technology of the entire company. Most of their competition, you'll see, will be dealing with a lot of other stuff. They don't really care about it. Maybe it'll be on the side of their website. Clear H2O really cares about the product, and that is huge. When it comes to customer service, their only focus is these gels and a variety of other stuff. Another great thing is their diversity of products. As I went through the list, they cover every area of enriching animals, which is huge for customers. Customers love to see that. Long shelf life, Clear H2O products last 12 to 18 months without refrigeration. As most of you know, in the labs, there's not a lot of refrigerators lying around, not a lot of space. So it leads to huge advantages for companies and labs. And the last thing, which I think is the most important, is evidence to back up products. People who work in labs tend to be more on the stringent side, which is great because no one wants to not be stringent with federal money. But it's really important that Clear H2O has evidence to back up their products because stringent people love evidence. And that's how they make their decisions while shopping. So actually, they just revamped their website last week to include data right beside the product on the website, which is very unique and very attractive to customers. And that's also where I come in, the collaboration. So customers have been wondering how long these mice can last off these gels solely without any supplements. Current studies had only gone out to 7 days, so we decided that I would take one out to 31 days and see how it goes. So we had 3 groups of C57s, which were actually bred right up at Jackson Labs, which is another cool main collaboration, and of 12 in each group. So we had the control of standard food and water, hydrogel group with standard food, and the diet gel 31M group, which is actually a product not even fully out yet with standard water, and just monitor their weight and overall health for a month. And this is what we saw. So on the bottom here, we have days going from 0 to 31, and the y-axis is body weight in grams. So the top here, we have the control in black, and the 2 gels, which did seem to lag a little bit. However, hydrogel significantly, looking at statistics, it wasn't that statistically significant. And that just has to do with random variability of weight, also how close the axis is. However, what we did see is with the diet gel 31M group, it definitely was a laggard. You can see all the stars of significance right here, indicating the probabilities. And we could tell that there was a clear gap there, and we were wondering why. So what we kind of took from that is just that we needed to make a slight change. Well, they actually decided that on themselves. I didn't take a part in that. However, I did find it very cool that 3/4 of the way through my study, they had already made a change in the composition of their product. They switched how much was kind of the hard diet and how much was hydration, and it has already been effective. I already tested it in some mice, and they seem to love it just as much as the old product, which is great. And with a hydrogel, as I saw earlier, there's not really a significant difference. And another huge thing, weight isn't the same as health. There definitely is a huge correlation there, but they're not the exact same. We did, however, monitor things like coat and behavior, and those, we couldn't differentiate between the groups, which is a great sign, showing that the mice were still healthy off all the products. And another thing, as you can see here, a very abnormal mouse. Fangs are a little out of control there. We did not find that in any of our mice. It was thought that maybe because we were just feeding them gels, that they wouldn't have anything to grind in their teeth, but that was not the case. We had a couple independent people come in and check them out and do some randomized groups, and nothing popped out. So in general, what I thought was so cool about this program is just kind of getting to understand the Maine biotech dynamic, because it's not something you really think about as a kid here. In school, K through 12, you never really deal with business or anything like that. You don't deal with industry. So it was really cool to experience it firsthand and to deal with these collaborations in Maine. Because once again, as I said, you don't really think about that. And the entrepreneurship aspect of this was just very cool. As I mentioned, being in the startup atmosphere is just something that's very exciting, and everything's going on at once, and it's a lot of fun. So in general, I'd just like to thank Dr. Bilski, obviously, Jordan and Henry for running the program, Jay Palmer, who's right here, happens to be my judge, and all the sponsors a ton. So thank you very much.
So we have time for a few questions if anyone has Any?
That was a great presentation, and I may have missed it, so I apologize. Was there any difference in the amount of gel consumption between the hydrogel and the diet gel, and is there any way of trying to ascertain whether it is a nutrient issue in general or maybe the specific type of diet that those animals lost weight?
That's a great question. We didn't actually measure consumption in the study, and there is certainly a difference in how much like diet gel and how much hydrogel they consume in a day. That would have to be something that we investigate with a different consumption study, and maybe perhaps look at dietary stuff. We didn't exactly look at nutrients in the study, just overall health.
Does anyone have any more questions?
Very nice. In the gels that you would like to use for analgesia, and in terms of pain relief, have you thought about how you're going to address the issue of dosing and outcome measure in terms of when you get to the point of sufficient pain relief?
Well, that was kind of what the grant is for. We're making the grant in order to get the funding to do those studies, hopefully here at UNE, which will be really cool. So we haven't thought a ton about it yet, but I mean, it's all stuff we can work out in those studies.
Do you have any thoughts about that?
The dosing?
Yeah. In other words, one of the things you're looking at is that I imagine these are so tasty that they would eat them ad libitum. Is that right? They would essentially could overdose on them.
They could, but I mean, it's just like humans with pills. You're just trying to, I mean, mice in general will eat about the same of the gel. So that makes dosing not that complicated.
Will eat about the same. In other words, they will stop eating even and not finish the packet?
Yeah, they do eat at about the same rate. They don't just go for it. Like we did discuss about that because like with the gels, you wondered, okay, were the standard ones just obese and just like eating as much as they want, or so that is an interesting argument. But we feel that they eat about the same.
Yeah. Okay. Thank you.
All right.
Thanks. That was a great presentation. I'm just interested to know a very, very basic thing, which is you can no doubt design the gels to have differing viscosity and different rates of dissolution. Does that ever play a role that the viscosity of essentially biting and gnawing on a gel consumes calories that just lapping up a liquid water would not?
Oh, I definitely imagine that. It's the same thing with humans. They say like apple and celery is negative calories. I can certainly imagine that happening. Though because we've done the weight studies, they've increased the amount of protein or calories so that the weights usually remain even. So I'm guessing they compensate for that. Well, that would be what would happen with those studies. They would definitely compensate.
About the weight, could you engineer a less viscous or less resistant formulation to burn fewer calories just in the act of taking in the water?
Yeah, you definitely can play with the viscosity of the gels. Usually, it kind of just works out with the nutrients, but I'm sure that's something they could definitely look into.
And we have time for one last question, which I think you wanted to ask.
So follow up on Bob's question on the body weight. And you basically have the answer right there from your graph, right? So maybe kind of link the control on the standard food and water. If that is, let's just say that is standard body weight and composition.
Mm-hmm.
And if you're under that with the first gel product that you tested, are they overeating that?
No.
No, so that basically answers Bob's question that they do limit their intake. So if you put medication in there and you know what the general intake it is, could you calculate out blood levels?
Yeah, definitely.
And could you verify that with analytics?
Mm-hmm.
Is that what you're going to do in the seed grant?
That is the hope and the awesome part about having the pharmacy department in Portland and then the behavior group here.
Yep, exactly. Okay, well, thank you very much. We're going to switch gears now. I should have mentioned all of the speakers are going to be judged today with a distinguished panel of judges, and so it's an added pressure, but we're going to have a little award ceremony at the end as well. And part of it is good questions from the audience and how they respond to that. So thank you guys for being interactive. Our next speaker is Rebecca Bind. She's working with Mike Berman, who's in our psychology department. And her project is entitled Effects of FA Inhibition on Fear and Memory and Insight into PTSD. And you're going to be a senior at Skidmore? Skidmore College in upstate New York.
Okay, so thank you, Dr. Bilski. I'm Becky, and my project was on the effects of FA inhibition and fear and memory and insight into PTSD. So what is post-traumatic stress disorder? Post-traumatic stress disorder is an incredibly debilitating neurological disorder in which people are completely overtaken by a traumatic event in the past. And so the criteria for this disease include having an event in the past that completely compromised one's life and reliving this experience through dreams, through flashbacks, and just being completely overcome by the trauma. And so it's incredibly debilitating. It affects not only the person suffering, but people around as well. And as you can see here, a large number of people suffer from it. So it's estimated that about 70% of people in today's society have undergone a traumatic event in his or her life, and around 20% of those people suffer from PTSD in total. And so as you can see here, around 50% of abused children will suffer at some point in their lives from post-traumatic stress disorder symptoms. And just another reference point, around 30% of people who come back from war suffer from PTSD. So as with any neurological disorder, PTSD greatly impacts the functioning of the brain. So there are two parts of the brain that are especially relevant in PTSD, and that's the hippocampus and the amygdala. And so these are the two areas of the brain that Dr. Berman studies in his lab, and that's how my project started. So the amygdala is the part of our brain that's responsible for fear and emotion, and the hippocampus is responsible for learning and memory. And so there's a circuitry that exists between these two areas of the brain, and they're connected by the perirhinal cortex. And so basically the relationship between the memory of the event and the fear that the event caused is what causes the post-traumatic stress disorder symptoms. And so studies show that people suffering from PTSD have reduced volumes of their hippocampus and amygdala. So if you look at these MRI, these are two different people. One is a control who doesn't have PTSD. And this person with PTSD, you can see this is the hippocampus right here. And compared to the normal person, there's a lot of atrophy, which means cell death. And also in PTSD, the amygdala becomes hyper-responsive. So when people become scared, someone with PTSD will have like a hyper-reaction to it. So this is just outlining the circuitry even more. The hippocampus, learning and memory, projects to the amygdala, which is responsible for the emotional stress, and projects all the way back. Okay, so in order to understand this circuitry a little better, it's important to know that there are two types of receptors that are especially dense in these two areas. And these are CB1 receptors and PPAR-alpha receptors. And so receptors have ligands that bond to them and induce a change in the brain. And so the ligands for both of these receptors are endocannabinoids. And so endocannabinoids are a family of neuropeptides in the brain located globally, but have diverse functions such as feeding, appetite, learning and memory, inflammation, mood. And so the three that are particularly interesting to us are anandamide, OEA, and PEA. And so anandamide binds to both of these receptors that I just mentioned, while OEA and PEA are more specific for this PPAR-alpha receptor. And so like any chemical in the brain, endocannabinoids need to be broken down at some point, and that's where fatty acid amine hydrolase comes in. And so this is an enzyme that breaks down endocannabinoids. And so the drug that we studied for my project are FA inhibitors. And so FA inhibitors, what they do is they basically prevent the breakdown of endocannabinoids, thereby increasing the levels of all three of these at the receptors. So a couple different things can happen when you use a FA inhibitor. And the first is an increase in endocannabinoids at the CB1 receptor. So the CB1 receptor is a G protein coupled receptor that's especially dense in the amygdala and hippocampus. And both endocannabinoids like anandamide and exogenous cannabinoids like THC, which is the main psychoactive component of marijuana, bind to this receptor and increase anandamide. And so what a FA inhibitor does is it increases the levels of anandamide at this receptor. And so what's particularly interesting to us is that with increased anandamide at this receptor and in the hippocampus and amygdala, anxiety levels usually decrease. And we know this because there have been studies done where rats have been given FAAH inhibitors. And so when rats are anxious, they'll do something called marble burying, which means they bury a lot of marbles in their ground. And if you give them a FAAH inhibitor, their marble burying will decrease, showing that they're less anxious. So the second result of FAAH inhibition is that you have an increase in endocannabinoids at this PPAR-alpha receptor. And so this receptor is thought to mediate learning and memory. So it's thought that if you have an increase in endocannabinoids at this receptor, you'll have an enhancement in learning and memory. So our study, we were looking to see how the FAAH inhibitors would work on fear learning and post-traumatic stress disorder. So we believed that FAAH inhibitors would lend to greater learning and memory for a fear stimulus, showing that increasing endocannabinoids in the hippocampus and the amygdala would have a stronger effect on enhancing learning and memory than on anxiety. So in essence, we believed that the effects on learning and memory would overrule the effects on anxiety. We were looking to see if there would be a dose-dependent effect. So we used 24 rodents and 3 different injections. 2 of them were the FAAH inhibitors, 2 different doses. One was 10 milligrams per kilogram, and one was 5.6, and then the vehicle, which had no drug in it. So in order to study fear conditioning, the assay, what we use is called fear conditioning, and it's an assay used to train subjects to learn to predict an aversive stimulus by pairing this aversive stimulus with a neutral stimulus. And by doing that, you gain an understanding of the relationship between the hippocampus and the amygdala. And so fear conditioning is done through a 3-day trial. And so the first day is called habituation. And basically what you do is you place a rat in either context A or context B. And as you can see, context A is square and it's dark, while context B is round and light. So 2 different environments right now. So that's the first neutral stimulus. The second neutral stimulus is that context A is wiped down with ethanol, while context B is wiped down with ammonia. So 2 different odors, and that's important because rats use their nose more than their eyes. And so on the first day, the rat is placed into either context A or B for around 30 minutes and receives a tone followed by a shock. And so the tone is the third neutral stimulus, and the shock is the aversive stimulus. Then on day 2, it's called context test. The rat is placed back into the same context as the first day for about 5 minutes and receives no tone or shock, but still has the environment and the odor. Then on the 3rd day, the rat is placed in the opposite context as the first 2 days. So if he was in A, it would be in B the 3rd day. And the floor is changed, and so it's a new environment, new odor, and no shock is received, but there is a tone during that amount of time. And so what you're measuring across these 3 days is the % of time the rodent spends frozen, because that would indicate that the rodent is afraid. So this is just kind of a timeline. So first day, 1 context, shock and tone. 2nd day, same context, but nothing. 3rd day, different context, and only the tone. So what you'd expect to see in a rat that was given no drugs would be a low freezing rate on the first day, because they had no idea what was coming. A high freezing rate on the 2nd day because their hippocampus is being used to remember what happened the first day in that environment. And then on the 3rd day, when they're put in a new context, there should be a low freezing rate because they don't recognize the environment, so they don't know what's going to happen. But when they get the tone will cue them that a shock is coming, so the freezing rate should elevate. And so on the first day, all the rats were injected with one of the 3 drugs 30 minutes prior to the assay. And this was to make sure that the drug reached its peak bioavailability. So these are our results. We were measuring % of time spent frozen across the different test days. And so what we found was there was no main effect of drug dose we used. There was a main effect of different test sessions, and there was an interaction between the dose and the test session, meaning the doses affected the different tests differently. And so looking more specifically, as you can see what we expected the first day, the habituation, the % of time spent frozen is relatively low, which is what we expected because they didn't know what was coming. On the 2nd day, you can see a huge decrease in the % of time spent frozen, which is not what we were expecting because we thought learning and memory would be enhanced, so greater freezing rate. And you can see that the 2 drugs pretty much acted the same. We did have a trend toward significance, but it didn't quite reach significance, we thought maybe our study was a little underpowered. So what you can see here is that with the decrease in freezing rate, we know that the drug was affecting the hippocampus and the amygdala circuitry because they weren't remembering as well as the vehicle. And then if you look at the third day, the novel context, very low freezing rate. This indicates that they didn't remember, which is good because it was a completely new environment. And then if you look at the tone fear, when they heard the tone, a huge spike in freezing rate, which is expected because they would associate the tone with the shock. And what you can see is the lower dose remained pretty close to the vehicle, but the higher dose disrupted freezing rate significantly, or almost significantly. And so if you compare these 2 days to each other, you can see that while this lower dose impacted context test, it didn't really disrupt tone fear. So we were thinking that this low dose only affected the amygdala hippocampus circuitry, but not the amygdala alone, while this high dose impacted both, so it disrupted the whole fear learning circuitry. Okay, so overall, we found that it seems the FA inhibitors show a reduction in context test freezing with drug treatment. And so just as a control, we know that at the lower dose at least, the rodents weren't affected by a FA inhibitor's antinociceptive properties because they can show to have like painkiller properties. But if you look at this, they still freeze, so we know that they felt the shock on the first day. And so how did the FA inhibitors work? Well, there is this conflict between are they anxiety or are they enhancing learning and memory? And so we thought they were going to enhance learning and memory, but it seems that they might have been working on anxiety more strongly. And so it's possible that these FA inhibitors were working more strongly on the CB1 receptors than on the PPAR alpha receptors. And so that could explain the lower freezing rate, because if the rats were less anxious, they wouldn't freeze as much because they wouldn't be as scared of the shock and the aversive stimuli. And so is this drug a viable solution for treating PTSD? You could say yes or no. If you look at the lower dose, you would say no, because while context test is disrupted, the tone fear is not disrupted, and so this wouldn't be desirable, because if you were to give a patient with PTSD this drug, or this dose of the drug, he or she would forget what the traumatic event was, but they would still have this implicit memory of what happened. So for example, if a war veteran were to come back from war and take this dose of drug and say a book fell off of a shelf and he jumped but had no idea why he was scared, that wouldn't be good because he would still have this implicit fear. But if you look at the higher dose, there is a decrease in both the context test and the tone fear. So this could be good because it disrupts the whole fear learning circuitry. So future research, we could probably add more subjects to the study to reach significance. We could insert the drug more locally, so directly into the hippocampus or the amygdala to make sure for certain that there are no antinociceptive properties happening, or we could do it in just the hippocampus and just the amygdala to see the difference in the fear learning. We could test the drug in different memory assays, not just fear learning, to see if it enhances learning and memory when it's not fear related. And we could administer the drug after day 1 instead of before, because if you think about a real-life post-traumatic stress disorder situation, I'd say a majority of people who have PTSD don't know going into it that they're going to have PTSD, so it's more realistic to test this drug afterwards, because not many people would take a drug prophylactically, and so that's the last thing we could do. So I just want to thank everyone in Dr. Bilski's lab, everyone in Dr. Berman's lab. This was an amazing opportunity for me. It was really great to be able to run my own project and see it go from start to finish. And thank you especially to all the sponsors who made this possible.
We have time for a few questions.
Hi, good talk. So I had a question. In your hypothesis, you described the treatment of FA inhibitor as enhancing learning and memory, and that being a potential treatment for PTSD. Is that correct?
We weren't, so if it enhanced learning and memory, we weren't looking at it as a treatment. We were kind of looking at it as an exploration, because for PTSD, you wouldn't want to enhance learning and memory.
Well, that was my question. So
yeah.
basically.
It was more of an exploration than this will be our treatment.
But it did disrupt the conditioning, right? Or at least it seemed to.
Right, which isn't quite what we thought was going to happen. We thought it was going to enhance learning and memory.
Okay. And then don't cannabinoids like THC disrupt learning and memory?
So we thought that there was a difference. So cannabinoids like THC, it's an exogenous cannabinoid, and FAAH inhibitors are an endogenous component, so they could work differently. And plus, THC works pretty much only at the CB1 receptor, while it's known that FAAH inhibitors also work at this PPAR alpha receptor, which show to have an enhancing effect on learning and memory.
All right.
Hi, this is a great talk. Very interesting study. I guess my question has to do with whether you think that giving the drug itself is a sort of cue, so that when you go back and you put the animal in the context, the animals that were given the drug are no longer on the drug, so that the conditions have really changed, and that's why you see less freezing. Does that make sense?
Can you repeat that again? Because I think I'm not quite understanding.
So in the conditioning stage,
Uh-huh.
when you're conditioning them, there are two groups of animals received the FAAH inhibitor, and that in and of itself could be a cue, or something that is different in that context. So that then
okay.
the next day you put them in the same context, but they don't have the drug, so they lack a specific cue. So that may be why they're showing less freezing. I'm just wondering if that's a possibility, or whether and how you might go about testing that.
So that's why we gave our third group a vehicle drug. So everyone got an injection. So the third group still had that stressor of getting an injection too. So they were all equal in that sense. Does that answer your question?
Well, I guess the issue here is that the drug is producing some central effect, so that it's changing the way the animal might feel,
Mm-hmm.
and so that may then be part of the contextual cue that it's using to pair with the fear. I don't.
Address that in one of your future directions. Which of your future directions would rule that possibility out?
So, thank you.
Sorry, I may not be clear in my question, but
So Ian's asking if the drug being there during training and not during testing changes the context enough. So it's a different context for them, right?
Okay.
Because they interpret the context differently.
So that's why I'd want to, I think this answers it, why I'd want to give the drug after day one instead of before day one to see the difference. Does that answer your question?
yeah, I think that's right. So you'd give the drug treatment also before placing them in the same context the next time. Is that right? Or
Train them and test them without the drug and give the drug after training. And that addresses that also.
yeah.
As long as the training and testing coefficient is the same, we would account for that. Right, can't account for that.
That's great. Thanks.
Question. Since PTSD is a chronic condition, I'm not sure whether if these results were borne out, and this could be a promising treatment, whether it would be just a few doses or a chronically administered drug. So my question is from a background of ignorance about regulation of the enzyme FAAH, is if you were to give an inhibitor, would the body respond by synthesizing more FAAH? Because sometimes in other contexts, if you give a lot of substrate, that induces the enzyme and it becomes more strong, and so enzymes themselves can get regulated in terms of their synthesis. I'm just asking out of ignorance, is FAAH an enzyme that could pose a problem that with chronic treatment would simply get induced to a higher level?
I don't completely know the answer, but I would guess that, as with anything else, too much of it wouldn't be desirable. But we didn't do a long-term study in this, but that would be interesting to do, because I don't know the long-term effects of it. This was only a 3-day trial.
Related question is whether, if on a genetic basis, are there strains of animals that seem to have a greater activity of FAAH, or is it something that is very tightly regulated across individuals?
I know that there are FAAH knockout animals. I don't know if different animals have different expressions of it, but I know that they can knock it out genetically.
We're going to have to move on to our next speaker to keep on schedule. Thank you very much, Rebecca.
Thank you.
Our next speaker is Adriana Urich. She is a med bio major, going to be a senior in Dr. Ling Tsao's lab. And she explores the interactions between the immune system and the nervous system. She's going to be giving an update on the research she's been conducting, I think, at least for the past year or so with Dr. Tsao.
As Dr. Bilski said, my name is Adriana, and I work in Dr. Tsao's lab where we focus on the immunological components of neuropathic pain. So just a little overview of what I'm going to be speaking about today. I'd first like to give some background information on neuropathic pain. I know a lot of our labs in the Center of Neuroscience study that, but I'd like to talk about more the immune system role in that. So I have the most recent definition of neuropathic pain up from the IASP. We know that neuropathic pain isn't really considered a disease, but more of a syndrome caused by a range of diseases and lesions that then manifest into different symptoms and signs. So there's multiple, the mechanisms underlying these conditions are multiple, and we don't really know a lot about them specifically. One thing we're commonly seeing is this presence of inflammation at the site of the affected nerve. This is where we see a lot of more recent studies in the area of neuroimmunology, trying to get a better grasp of what's going on in this sense. So I found this great diagram in a recently published article showing kind of what's going on between the communication in the immune system and the nervous system. So there's a key up here that'll help. But what I have here is an injured side, the dorsal horn of the spinal cord, and on the injured side. So there might be some periphery nerve injury over here versus the uninjured side. So normally we have these resident microglia and astrocytes, amongst other things, in the dorsal horn of the uninjured spinal cord. When there's an injury in a nerve or some sort of neuropathic pain going on, we see the cells become activated and we see proliferation. So the bolded red and blue are activated microglia and astrocytes. What's more important is we see this release of all these pro-inflammatory mediators. And this is contributing to this inflammation going on in the nervous system. And that's kind of what our lab likes to focus on. We want to understand these mechanisms better. I specifically, in the study I'm going to be talking about today, am looking at microglia. And microglia are essentially the macrophages of the spinal cord and the brain. So they're basically the first main form of the active immune defense in the CNS. Before I start with my study, I just want to mention a previous study Dr. Tsao was involved with, which actually led to a lot of the experiments we're doing in our lab. What she worked on was a study that looked at the mechanical sensitivity of CD40 knockout and CD4 knockout mice compared to the wild type in a post-L5 nerve transection model. We used the same model in the study I'll speak about today, which I'll go a little bit more into detail in my methods. What was seen was that on the y-axis here, we have the threshold force given to the mouse, and on the x-axis, we have the days post-surgery. The wild type is in red here, and the lower the threshold is basically correlating with a more hypersensitive mouse. So a wild type mouse we saw was very hypersensitive over the entire 21-day period. Well, I didn't work on this, but Dr. Tsao. In this experiment, that's what they had found. In this graph here, we have the CD40 knockout mice, and the green is the transected mouse. So over the 21-day period, we saw a decrease in they found a decrease in hypersensitivity over the entire 21-day period. And up here, the gray and the black are the sham surgeries, which essentially is a surgery performed that the nerve was not cut, so it's another form of a control. And then we see in the CD4 knockout, we also see this decrease in the blue. The red is the wild type again. So that's decreasing over time. And from this experiment, we want to know more about what's going on with the CD4 and the CD40 and why is that leading to this decreased hypersensitivity. So this poses the question, how can we further delineate CD4 and CD40 mediated mechanisms and involve the development of neuropathic pain? So for the model, to speak a little bit more about it, we use BALB-C mice, and we have this L5 nerve transection model. This diagram here has a few things going on, but what I'd like you to focus on is that from the L5 nerve, we actually do a transection here with the red zigzag mark here. And some of the mice are exposed to this transection, but some are exposed to a sham surgery. And like I'd said, surgery is performed, but the nerve is not transected. We have 3 types of mice: wild type, CD4, and CD40 knockout mice. CD4 knockout and CD40 knockout. Just to speak a little bit about CD4 and CD40 and why those might be important, CD4 is a co-receptor found on surface of immune cells like T helper cells and macrophages. And those are important with the communication with antigen-presenting cells. They essentially amplify the signal between them. And then CD40 is a co-stimulatory protein that's found on antigen-presenting cells. So it's vital for their activation. With these 3 types of mice, and after the surgery, I would pool the lumbar spinal cords, and I'd collect them on day 0, day 3, day 7, and day 14. We then would do cell isolation of the mononuclear cells, and then I'd take these cells and do flow cytometry to analyze them. So the way the spinal cords are pooled is into similarly treated mice. So L5 transected ipsilateral side would be pooled together for a certain time point, as well as L5 contralateral, and then we do the same for the sham. These are labeled with monoclonal antibodies CD11b and CD45. And here we have a little bit about what we would see after doing flow cytometry. So this would be what we'd see, and this is the cell population that we're going to be looking at here. All this here would be primarily debris. And then from this cell population here, we can target the microglia cells by CD45 in low expression and CD11b, so that would be our microglia population. And we also would look at cells that had high expression of CD45, and that would be infiltrating leukocytes. So now I'm going to talk a little bit about the results that we found. I'm going to start with a figure from the numbers of microglia from the lumbar spinal cords after the surgery in the wild type. And I'd like to explain the figure a little bit. On the y-axis, we have the numbers of microglia. On the x is the days. So essentially, this is day 0, day 1, day 3, day 7, and day 14. Within each one, like I said, those 4 groups that we pool the spinal cord into, we have the transected IPSI side, so that's where the nerve was cut. TX would be transected, SH would be sham, ipsilateral, contralateral. So each one of those for the time points. What we found in a wild-type mouse for the microglia population was a significant increase at day 3 on the transected ipsilateral side. And this was compared to all other groups at the same time point, as well as all groups all time points in the same treatment group. We also see a significant increase at day 7 in the microglia of the transected IPSI side. To compare to the wild type, compare the CD4 and the CD40 knockout mice to the wild type, the CD4, we did see an increase at day 3, again, on the ipsilateral of the transected. And we did see an even greater increase at day 7. The CD40, we have a lower N number, and unfortunately, we can't really have a trend going on right now and are trying to understand we'd like to add more data to understand what's going on here, so it's hard to say right now. But it's interesting what we found in the CD4 knockout. For infiltrating leukocytes, what we saw in the wild-type mouse, this is the same axis here for the X, and this would be the leukocytes. We saw an increase at day 7 in the transected IPSI side. The picture in the corner, again, is from the flow cytometry data, and that's where we would quantify this from. And now looking at CD4 and CD40 for infiltrating leukocytes compared to that wild-type graph I showed you, we didn't really see any similar increase going on along the time point. Again, CD40 is hard to understand right now until we get more data to add to that. To summarize what we basically found in the study, for the infiltrating leukocytes, we did see this increase going on in the wild type, and that occurred at day 7 of the the transected IPSI. Since we didn't see any similar increase in the CD4, CD40, this could suggest that CD4 or CD40 play a role in inducing the infiltration of leukocytes. The microglia, what we saw again in the wild type, was that increase at day 3 and day 7. When we looked at CD4, the largest increase happened at day 7 of the transected IPSI, and we're wondering if maybe CD4 plays a role in the early microglial responses. CD40, we still would like, in our future studies, we'd like to find out more about that and add more data to see if there's a trend, if a trend at all. So that's kind of where we're headed in our future directions. I'd like to thank Dr. Tsao and everyone in our lab. Holly Bolak, I'd like to mention her because she actually contributed a lot of time to this experiment as well. She went on to grad school. So she's not here today. Jen and Kyle helped out with the flow cytometry, and I'd like to thank the Center for Excellence in Neuroscience for putting on this event today and all the opportunities that we get here. So, yeah.
Does anyone have any questions?
So you were talking about the, you're not sure quite what's going on in the CD40 knockout mice. What are your thoughts about that? And where do you think the future direction could go with those?
Well, we breed the CD40 mice here, and a few different experiments are using them, so I don't have a high N number for that compared to the others. What we do think it's important based on the study of the hypersensitivity, because we saw that decreased hypersensitivity going on. So we'd like to know what is going on with microglia, because we have like 2 different things, like some with high populations, some with low, so it's a little weird there. But we'd like to add more data and understand if there's a trend. I think it's important. CD4 showed that maybe it's important early on, so I feel like CD40 being part of the antigen-presenting cells, I feel like that must have some role. I hope that answered your question. Yeah.
The CD40 knockouts again. So they did seem like they were sort of disordered compared to your wild type, more variable, and also maybe more immune cells. Do they have a phenotype that would suggest that inflammation or the similar?
Phenotyping, did you say?
Do they have a phenotype that would suggest inflammatory issues, increased inflammation, higher immune cell count?
A phenotype for a type of mouse, I'm sorry, I'm not sure.
Do they have a
for CD40 knockout?
Symptoms, I guess you would call it, of the CD40 knockout.
I don't do any for like a behavioral area or behavioral testing.
Anything.
Well, I haven't done any behavioral testing. I don't know if it will answer your question saying how they, as in the previous studies with the decreased hypersensitivity, they showed more decreased hypersensitivity than CD4. I don't know if that's I didn't do any behavioral studies like looking at that area, just the cells. I just did flow cytometry looking at cell populations. So
sure.
We're just we saw some high numbers, but some really low numbers, so we don't know why there was a variability there. Yeah.
And then it did look different from the wild types. So were the wild types littermate controls, same strain, different litters?
Oh, they're all BALB-C mice. They were done over a long period of time, so they're not the same litters. We had done it throughout the entire year, so we just keep adding data to it. So they're not from the same litter or anything.
Any more questions?
I had a question that I think maybe comes from the opposite point of view of the one just asked. You know, if you say to a pain clinician, CD4, the thing that a lot of people will immediately free associate with is pain related to HIV/AIDS disease, where the definition of AIDS, at least the original CDC, was posed in terms of absolute CD4 white cell count. Yet in HIV/AIDS, the most common pain syndrome is a neuropathic pain, although the particular kind of neuropathic pain can vary quite a bit. And I'm trying to reconcile the loss of CD4 positive white cells and concurrent neuropathic pain with the findings you've presented. do the animals that are CD4 knockouts lack CD4 throughout all cells, white cells, glia, etc.? They just don't have that expressed?
Yeah, they are CD4 knockout mouse. Interestingly, how you're talking about the AIDS relation, HIV, we actually have an experiment going on, Dr. Sau's been very involved with, and it's a model, it's a MAIDS model, so kind of mimicking what's like HIV in a human, and trying to study the neuropathic pain side and what's going on in the immunological sense in the mouse. So we do have a study going on in our lab in that area, so it's kind of an interesting point that you brought up, that the relation between that. So that's another study we do have. So.
Thanks.
Okay, well, thank you very much, and we'll move on to the next speaker. The next speaker is Ian Imbert. He works with Dr. Tamara King, who's an assistant professor in the College of Osteopathic Medicine. Ian's going to give us an update on some of his most recent research around osteoarthritis pain and a rodent model and some of the exciting discoveries they've been coming up with. Ian?
All right, well, hello everyone. I'm Ian Imbert. I'm representing Dr. Tamara King's lab. I'm going to be taking you through the basics of osteoarthritis and some of the pain that's associated with it. And then we'll look at how osteoarthritis is modeled at the preclinical level. And finally, we'll try and figure out a better way to treat osteoarthritic patients who are NSAID resistant. So why do we want to study osteoarthritis? Well, according to the CDC, there was roughly 27 million US adults who suffered from the disease in 2005, which means that nearly 10% of the entire population suffered from osteoarthritis that year. Osteoarthritis is usually associated with a decreased quality of life because subjects experience decreased mobility, which can lead to depression, physical disabilities, and increased healthcare costs. It's also believed that as the prevalence of life expectancy and obesity prevalences continue to rise, as will the prevalence of osteoarthritis. Furthermore, the only current effective treatments of osteoarthritis aim at relieving symptoms without having any curative effect of the disease. So while non-steroidal anti-inflammatory drugs, so your aspirin and ibuprofen, may be effective for the casual athlete who experiences joint discomfort while playing tennis, but for subjects who experience more ongoing form of osteoarthritis, there's a dilemma of dose effect because of adverse side effects. And due to the lack of curative treatments and diminished quality of life, many NSAID-resistant osteoarthritic patients have joint replacement therapy as a last resort. And while joint replacement therapy may be effective, it's expensive and invasive, so the goal at the therapeutic level is to produce novel, less invasive therapies to treat NSAID-resistant ongoing joint pain in patients with advanced osteoarthritic pain. This image shows pathological changes between a normal knee and an osteoarthritic knee. As you can see, there's certain cartilage loss in the knee joint, there's absence of key lubricating agents, and there's abnormal bone formation known as osteophytes. I thought this radiographic image showed that osteoarthritic pathological changes are also characteristic of a narrowing joint space. And although these are characteristic pathological changes of an osteoarthritic knee, it's important to note that pain reports do not always correlate with pathology in osteoarthritis. So there's generally two different types of osteoarthritic pain. We have early osteoarthritis characterized by movement-evoked pain and usually described as a sharp stabbing pain. And then there's advanced osteoarthritis characterized by ongoing pain, which is usually described as a constant throbbing or burning pain with occasional bouts of stabbing pain. So this advanced osteoarthritic group is the specific target group that we're looking at because they're NSAID resistant, and we're trying to produce novel therapeutic drugs to prevent them from having to have that invasive knee replacement surgery. Interestingly, until recently, there was no preclinical model of advanced osteoarthritic ongoing pain as experienced by patients with advanced OA pain. Early osteoarthritis, however, has been modeled, and it's usually modeled by intra-articular injection of the drug monosodium iota acetate. So monosodium iota acetate is injected into the intra-articular joint, and as early as 7 days later, osteoarthritic symptoms are seen as verified by weight asymmetry. This is a weight-bearing apparatus, and this is how we measure weight asymmetry at the preclinical level. This figure shows 3 completely different MIA doses. However, interestingly, they all show similar, non-significantly different weight asymmetries. But remember, we want to measure ongoing pain, and there is no model of it, so that's what we're specifically trying to target. So before we can develop a model of ongoing pain, we need to try and figure out a way to measure ongoing pain. If we were in a clinic, we could simply just give the patient a drug and then ask them how they feel afterwards and see if the pain was taken away. How we do it preclinically is we administer a local anesthetic, something known as a peripheral nerve block, where lidocaine is administered into the intra-articular knee joint. And then we have to try and figure out what exactly is the rat thinking, because we can't just simply ask them, how they're feeling and see if the pain's taken away. So one way we can do that is by a behavioral assay known as condition-place preference. The condition-place preference is one way that we can talk to and monitor the emotional states of rats to try and figure out how they're feeling after we've given them that local anesthetic. The condition-place preference assay uses different sensory cues to try and monitor the emotional states of rats. There's different textural, visual, and smell cues. As you can see in pairing chamber 1, there's a smooth floor, whereas in pairing chamber 2, there's a textured floor. In pairing chamber 1, there's white walls with black stripes, whereas in pairing chamber 2, it's solid black walls. And then also, in pairing chamber 1, there's a strawberry scent, whereas in pairing chamber 2, there's a vanilla scent. It's important to note that the middle chamber is unpaired and is considered neutral. So the condition-place preference is a 3-day assay. Day 1 is actually 13 days after the initial administration of the monosodium iodoacetate into the intra-articular joint cavity. It's considered baseline day, and it is important to make sure that there's no naive biases in the rats. So the rats are placed in the chamber for 15 minutes and are allowed to freely roam around, and then taken out. And the following day is known as conditioning day, and is broken up into 2 different parts. In the morning, the rats are administered intra-articular saline and are restricted to pairing chamber 2, so this door right here would be closed. And they're restricted to pairing chamber 2 for 30 minutes. They're then taken out, and 4 hours later in the afternoon, they're administered intra-articular lidocaine and are restricted to pairing chamber 1 for 30 minutes. They're then taken out, and the next day is testing day, and once again, they're placed in the chambers for 15 minutes. This time, there's no drug administered, and what we expect is that this time, the rats are no longer naive. So what we predict is that the rats will spend more time in the chamber paired with a peripheral nerve block. So in this case, it was pairing chamber 1, because the rats are going to remember that experience of pain relief. What was found is that the peripheral nerve block 14 days post-MIA injection induces condition-place preference. However, only in the highest previously untested dose of 4.8 milligrams MIA, which indicates that this treatment group only was experiencing chronic ongoing pain. So in review, we have our 2 types of osteoarthritic pain: early osteoarthritis characterized by movement-evoked pain, and advanced osteoarthritis characterized by ongoing pain. We have 3 different MIA doses, all which produce similar, non-significantly different weight asymmetries. However, when you take those 3 treatment groups and you condition them in the condition-place preference assay, despite the similarities here, you can see there's apparent differences here where only the highest previously untested dose of 4.8 milligrams showed condition-place preference. So we have our 2 doses, the 1 and 3 milligrams MIA, which have been modeled as early osteoarthritis. And for the first time, we have a dose of 4.8 milligrams MIA that has been modeled as ongoing pain or advanced osteoarthritis. So what we're looking at now is we're trying to figure out, so what's this mechanistic difference that's causing these behavioral changes between early and advanced osteoarthritis? we think that a key difference is the central processing and pain. So in our lab, we are running immunohistochemistry on sections of the spinal cord to test to see if a marker of central sensitization can be found. So in order to test for central sensitization, a normally non-noxious palpation was performed on the knee joint across all treatment groups. If the palpation induces FOS, a protein that is only found in the spinal cord upon noxious stimulation, this would be suggestive of central sensitization. So these images are showing the spinal dorsal horn on the ipsilateral side of these osteoarthritic mouse. And I then proceeded to count FOS by using this key here of the different Rex's lamina. And it's important to note that there were 3 rats per treatment group and 10 slices were averaged per rat. And so this figure shows the quantification of the FOS expression across the 3 treatment groups. As you can see, there's no significant difference in FOS expression between the saline and the 3.0 MIA dose. However, when looking at the highest dose that's been shown to mono-advanced osteoarthritis, there's a significantly increased expression of FOS when compared to both the saline and the 3.0 milligram treatment groups. So in conclusion, we were able to model advanced osteoarthritis for the first time preclinically. And we plan to further substantiate our hypothesis of central sensitization by further radiography and immunohistochemistry techniques. I'd like to thank my PI, Tamara King, and her collaborator, Frank Parecka, my laboratory manager, Josh Havlin, and coworkers Adam Sarcheski, Jenny Cormier, and Ian Pelletier. I'd also like to thank Ed Bilski for putting on the Capstone Symposium and letting us present our data today. And at this point, I'll open it up to questions.
When you're quantifying the FOS on the IHC slides, what did you use as a positive control, and what technique did you use to actually count the positively stained cells?
Okay, so for the positive control, would that be the saline rat? Or
If you know what the FOS is going to look like,
yep.
how do you know what a positively stained cell is going to look like?
Oh, so you want to know what does a FOS cell look like?
Yeah.
So here we see, this is a FOS cell here. These are basal levels, so in the saline. And then, unfortunately, it's a little bit pixelated, but you can see that you can even visually see the significant difference in FOS expression. So normally you see high FOS levels in Rex's laminas 1 and 2, and then also in deep lamina, so in lamina 5 as well. So right here, I find that you find most FOS in either 1, 2, or 5.
And did you use any different kind of tissue type to make sure that that's what you were seeing as opposed to a background?
Well, right now we have knee joints. We just slice them, and we're going to be performing CGRP staining on them. So that would be one other way to substantiate this data as well.
So did you always give the lidocaine in the afternoon and the control in the morning?
Yep.
Yeah. So I would just mention that there are circadian issues around pain perception.
Sure.
And so that's something you may want to think about in the future.
Mm-hmm.
The other question would be, if you look at your effects on percent shift upon weight bearing, you saw an effect of across the dose range.
Yeah, it does. There is, I guess you would say, higher asymmetries as the dose is increased.
Yeah,
But
yeah,
it's
they were
not-
slightly high, but you certainly got effects on
Yep.
percent shift.
Yep.
Whereas that was not manifest at all at the lower doses in CPP. That is the perception of pain. And so here again, when did you when you look at the percent shift in weight bearing, what time of day do you do that?
I can't say that it's consistent. I think that It's a long test. We do 3 trials per rat, and if I have 24 rats, it might take me 3 or 4 hours, so I might start at 10 and finish at 2.
Okay. You might want to just look over time and see what kind of variability you're seeing in that group, so that you can address that question, because it's interesting that there was no evidence of any kind of for the rat in terms of perception of pain in the CPP design at the lower doses. The major effect you got was certainly relief of pain, and that was seen at your higher dose.
Well, I think what's interesting is that you wouldn't expect to see any perception of pain in the early osteoarthritic patients or in the early osteoarthritic doses, because they're only experiencing pain on palpation or movement. So after we give them the lidocaine and place them in the chamber, they're not experiencing pain. But the ongoing rats are experiencing that constant pain, so when you administer the peripheral nerve block, that pain is taken away. So you're not actually taking away any pain in the rats who are experiencing the early osteoarthritis, because they're not experiencing pain before, because the knee joint hasn't been palpated.
Hi, thank you. That was an excellent presentation. As an outcomes researcher, I'm very interested in the literature for osteoarthritis, because the placebo effect has been shown to be huge. And randomized studies for people who undergo knee surgery, placebo surgery actually is as effective, if not more effective, than real surgery. And I'm wondering, given that that's such an important component, the placebo effect for osteoarthritis in people, how you might be able to try to use an animal model to get at the placebo effect with animals. Is that and if not, how do you apply this across species where that is such a compelling component of people's perception of pain?
Yeah, that's a really good question. I don't have much clinical experience. We are collaborating with a clinician right now, and she's mentioned that the placebo effect can be devastating for research at the clinical level. I'm not sure. I know that I don't believe we control for it. I think that I don't know how you can test it. It's really hard to communicate with the rats unless you were to administer saline in the morning and maybe saline in the afternoon and see the effects of that. That could be one way you could test for placebo. Other than that, I'm not really sure.
Okay, well, thank you very much. So our next speakers, this is speakers plural, are Evan Shuris, who is just graduated from Kennebunk High School, and Trevor Luck, who is going to be entering his senior year at Kennebunk High School. Evan did the program last summer and was also, played an important role in tying the University of New England with the Kennebunk school system as it expanded its outreach efforts. And what was kind of nice was that he found someone who was also interested in this. And so we have a legacy in Trevor, and we hope to continue this as some continuity from year to year. And these guys have also volunteered not just to do outreach in the Kennebunk systems, but go to the other school systems as well. So it's kind of amazing to think, from a high school perspective, taking on something as big as this, and then going and presenting as a tag team, which Evan has done at least once before, the prior summer. So without further ado, Trevor and Evan.
Okay, awesome. Thank you, Dr. Bilski. As said, my name is Evan Shuris. I'm actually going to be an undergraduate at the University of Connecticut in the fall.
And my name is Trevor Luck, and I'm a rising senior at Kennebunk High School.
We're here to talk to you today a little bit about K-12 outreach in the neurosciences and how we can play a role in the K-12 system, in addition to playing a role in the main biotech system. So just a little background on what we do. Our program is composed of different modules, which are core themes on which we actually present on. The modules are composed of interactive activities, and they range in age groups between K through 5, middle school, as well as high school. These modules are presented by UNE faculty, undergraduates, medical students, all the way down to high school volunteers. And this is useful because it gives students an opportunity to look at all the ranges from different perspectives as far as their high school peers all the way up to university faculty, so they get to see the whole range of what they might encounter.
No, I'm going to use this term STEM, and I'm sure most of you know it, but for those of you who don't, it stands for Science, Technology, Engineering, and Math. And what has been found is that students at a very young age have this innate interest in STEM. They're fascinated by science, And over time, as they progress through the educational meat grinder, that interest dies out over time. And the problem is that Maine in particular, as a state, is falling behind in STEM. This is in part due to the fact that our educational systems aren't as strong as other states in STEM. For instance, UNE is actually the only medical school in Maine. You don't have the option of becoming an MD or a DO as a student in Maine. You can only become a DO. Other states have both options. And as a result, more and more students are leaving Maine to pursue STEM careers elsewhere. So, as a whole, Maine is falling behind in STEM. And as you can see by this graph right here, this quantifies the fact that the interest dies out over time. Right here on the x-axis, this represents different student groups by grade level. The y-axis represents student population. These blue bars right here represent the total amount of students in the educational system, whereas these pink bars represent student interest in STEM. So you can see it dies out over time. In 8th grade, we're using Math Proficient as a surrogate for STEM interest. In 8th grade, 29% of students are essentially interested in STEM. Only 23% of college students enroll with a STEM major. And only 40% of them will actually graduate in 6 years with that major.
And this STEM deficiency issue is actually recognized at the federal level through the National Institutes of Health. They actually have designed a whole blueprint for neuroscience education that ranges from the doctoral level all the way down to the K-12 system. And they actually believe in choosing different methods to present science education. They believe in choosing online resources, or as we've also chosen, hands-on activities to bring new students into the science field and keep them interested long enough to actually declare and maintain a STEM major.
So Evan, how does UNE offer a solution?
Well, I'm glad you asked, Trevor. This is a list of our current modules. We believe that students in our system, we want to actually grow with them and have them grow up with us and grow out with us. So we want to have them grow up with us as we follow them along through the K-5 system all the way through middle school and high school, showing them different modules and as they increase in complexity and increase in information, going from just maintaining their interest in science at the K-5 level towards actually learning information about sciences as they go farther into high school. We actually grow out with them as well by teaching them different areas of science, using neuroscience as a lens to teach all the ranges from clinical neurology and psychiatry to cognition, all the way to basic neuroanatomy.
So we have certain goals in mind. First and foremost, we want to expose kids to fun, interactive science activities which will help foster and maintain a passion for STEM. Working with their interest in STEM that's already in place, we want to really work with this and get them interested in science so that they stay interested not just in middle school, not just in high school, but throughout college into their future careers. And with this, we also aim to have our curriculum supplement the current science curriculum in school so that what we're teaching them isn't just some random topic often somewhere in the land of neuroanatomy that doesn't really mesh with what they're learning in Intro to Bio. We want, our goal is to really make that work with what they're currently learning, as well as exhibiting what UNE has to offer as a well-rounded university. We're getting the name of UNE out there. We're marketing UNE, so to speak.
So what did we manage to accomplish this summer? Well, we managed to look at more of the actual structure of our program, to look at more of how can we actually present what we're doing more efficiently, and so the students get the best experience. We did actually do some more module development, so we are actually offering a new, brand new module that involves a new area of science for us, so that's also exciting as well.
So first and foremost, the neuroscience and physics activity that Evan and I worked on. We paired with Dr. James Vasenka in the Department of Chemistry and Physics to develop a helmet drop activity for our traumatic brain injury module. And what this is essentially, we're bringing the idea to kids that it's good to wear a bike helmet or a helmet of any sort when performing dangerous activities, potentially dangerous activities, you know, skiing, biking, so on. And what we're doing is we're showing them how this works. So we take a water balloon of a certain mass, we drop it, And we drop it in increasing 1-foot increments until the balloon breaks, setting a certain height threshold. And then we do the same thing over with the balloon protected by a helmet. And if, provided everything works out, the height threshold should be higher. And with the mass of the helmet and balloon and the height thresholds in mind, we're able to calculate the impact energy of the balloon and helmet complex to bring data to the students so that we can quantify. We can show them that it's scientifically proven that it's safe to wear a bike helmet, rather than saying, wear a helmet, believe us. And this also exposes students to areas of science that they may not have yet experienced. So showing kids in an AP physics class the neuroscience part of this physics activity, or showing kids in a bio class the physics part of a bio-related activity.
So another issue we ran into was we asked teachers, what do you think of our program? They say, it's great. You guys show up, you guys teach neuroscience, but you only come once and you don't actually fit in with our curriculum as well as we'd like. So we've developed a whole new curriculum design that aims at actually outlining what we're doing and showing the basic intro to the activity, the learning goals, the activity itself, and a basic way to assess student progress. And this is useful not only for our side, for better being able to present what we want to offer, but also it helps K-12 teachers prove to their administrators that what we're doing is worth their time and worth their students' time.
Evan and I also are spearheading training videos for the outreach program. So currently the issue is that training for our volunteers is a bit scattered and non-standardized, so that the product we're producing sort of varies from school to school. The way we present the neuroanatomy module may be different at Kennebunk High School from how we present it at Sanford High School. And so to make our products better, we need to standardize the way that we teach our volunteers our material so that the product is consistent. And so with this in mind, Evan and I ran through an introduction on how to, the TBI module. We made a 5-minute video explaining how to teach the module. I ate some glucose. We dropped a melon. It's both an entertaining and educational video. And this will also reduce the time consumed that it takes to actually teach our volunteers to how to do these modules.
So this is great. We've got a whole set of brand new volunteers that are well-trained and ready to go. But how do we prove what they're doing is actually effective? Well, we utilized an on-campus summer camp called KidFit that's aimed at teaching kids more about physical education. They actually use interactive activities in their own right, so it was a great choice for us to use. We presented their students with a basic traumatic brain injury and reflex module that showed them a little bit about the basics of neuroscience. And our whole goal here was to not only prove a quantitative approach as far as is this effective, but also just to aim students to see if they can actually understand what we're trying to present and to see if it's actually going to push them more towards science careers.
So what's next? Our short-term goals in mind right now are obviously to fine-tune the module offerings, make our product both more fluid so that it works with schools to better implement them into their curriculum models, and also to better educate students. We want to develop grant applications. With more funding, we can improve the overall status of this program, as well as branching out to more schools so that we increase our area of effect, and incorporating more faculty here at UNE to broaden the module offerings. Not only do we want to involve Dr. Vasenka, but more faculty here at UNE.
One such grant that Trevor's referring to is a SBIR grant, so Small Business Innovation Research Grant funded by the NIH. This grant is actually finely tuned towards what we're doing. It's actually aimed at K-12 neuroscience education and innovative ways to present that. It can reach in excess of $250,000. And after seeing what kinds of groups have won these programs, we seem to stand a very competitive chance, having already developed a very good program.
And as for our longer-reaching, or more farther-reaching goals, we want to work on these big NIH grants that Evan just mentioned, develop more corporate partners to help stimulate the overall growth of this program, as well as developing a business model for this to train local teachers how to better implement our curriculum design. and broaden the program to reach out more to northern Maine, because as of right now, we're sort of stuck in this little pocket that is York County, and the state of Maine is falling behind in STEM throughout, not just in the south, but through the north, the central Maine, throughout. So if we increase our area of effect, we can help the state as a whole. As well as developing connections with area biomedical centers, which include but are certainly not limited to Jackson Laboratories, Corning Life Sciences, UMaine Graduate School of Biomedical Sciences.
And so to reflect on the program, this has been a fantastic opportunity. It's really opened my eyes to what science and medicine can be, and it's really given me an idea of what I might want to do with my career. I'm thinking more of how to combine business and science and not only be a clinician as a physician, but being able to tie in some sort of biotech company into it. So getting the best of both worlds.
Yeah, and as for me, let me preface by saying that being only a high school student, I never imagined I would get the opportunity to do all of the wonderful things I've done as part of this program, from the lab experiences I've had to the seminars with big names in the biotech industry to lectures on pharmacology, cell biology, and neuroanatomy. This program has really broadened my horizons. Beforehand, I only saw myself in the patient care side of things as a doctor, but now I'm looking more at the business and research side of medicine. So thanks to this program, my horizons have definitely broadened.
We also want to broaden our thanks out, especially great thanks to Dr. Edward Bilski for actually coordinating this program and making it what it is, as well as all the professors involved in giving us lectures, which were fantastic, as well as all the different sponsors of the program, especially the Michael T. Goulet Foundation. And we'd also like to thank the Bilski Lab for being so warm and inviting, and as well as the two people who were greatly involved in coordinating our program, Henry Fairland and Jordan Falloon, who, I don't know if you read her name tag this morning. She's officially a member of the College of Osteopathic Medicine. And Jordan, we'd like to give you a great send-off here. Alex, you show our appreciation for everything you did for us this summer. And with that, we'll take questions.
Oh, we're probably going to need to, yeah.
Nice job. It was an interesting talk. The one question I have is, do you have any plans to track any of the schools or the kids that you've brought through this program to see if you increase the STEM percentages in those areas?
That's been discussed, and it's an issue that is obviously going to take a great amount of time. I mean, we work with students in all different age ranges, but right now we're looking more towards the short-term quantitative ideas. So did we actually influence them in the weeks after? Were they more interested in science? But it's actually a great idea. We'll actually look more into how they actually perform later in life and see if they declared STEM majors. So.
Yeah, I also think it's a terrific program that you've accomplished. And in addition to the basic science and entrepreneurial dimensions, you've also presented a public health dimension, in a sense, that by having a hands-on experience that illustrates what are the benefits of wearing a helmet, you can teach better, that it's good to. But I'm wondering whether you're planning to consider other behaviors that would pose risks that are so prevalent, you may think of them as a public health issue, in the younger population. For example, I'm just pulling these things out of thin air, texting while driving. If you had a driving simulator and then someone who was asked deliberately to text, and you could show how their performance was impaired, or some other type of intervention that would target specific public health problems in the age groups that you're addressing.
So as this program grows, we're definitely going to implement some of those issues into mind, and that is a great point that you've brought up. So yes, just in general, as this program grows and more modules are developed, we are going to include issues such as that in them.
Actually, you're closer, then I'll go over to you, Shelley.
Your model seems to suggest that the key to engaging more students to pursue STEM-related careers requires that we help students to have and help teachers to give students problem-based experiences in the classroom around topics like neurobiology that are really exciting to students. I'm wondering if you have considered, you've talked about scaling up. I wonder if you've considered partnering at all with the Department of Education to bring greater visibility to some of the tools that you have.
Well, we actually haven't. That's actually a great point. That could be a great opportunity for us. I mean, we're looking for more opportunities to grow and opportunities to present new materials. So that could be a great connection.
So wonderful presentation. I want to build on Dr. Carr saying that it has enormous public health implications. And again, when, in the spirit of partnership, wondering two things. One, if you're considered partnering with our School of Public Health, and what's, bringing in a real interdisciplinary model to what you're doing. And secondly, the fact that children are dependent on their parents or caregivers, and whether you've thought about adding a component of outreach to the parents as well to really reinforce what it is you're teaching your students.
First and foremost, with the partnership idea in mind, I don't think that has actually come up before, but it's definitely a really great idea. Yeah, and I'm sorry, what was the second part to your question? Parents.
Actually, that's another great opportunity for us to have. I mean, a lot of it comes down to how do we actually reinforce the information we present. And most often, parents are the greatest influences on their children's lives. So even doing something simple as giving them a take-home kind of thing where they can show to their parents, what did I do today in school? And having the parents follow up on that can be just a great motivator for students to actually retain that information.
I'm going to make a quick comment with these guys, because I've been struggling with this modules and calling them vertical and horizontally integrated. And you guys just within, I don't know when you came up with the idea, but grow up and grow out. That's a fantastic moniker, I think, that we're going to be able to use. And neurons to neighborhoods, that we kind of stole. But this one I think is original, at least applied to neuroscience and the outreach. So I congratulate you guys for coming up with that. And also just as a follow-up to your question on the education, this fall we're going to reconvene a broader group across the university, including our Department of Education, mathematics, and many other health professional programs, to try to better coordinate all of the outreach efforts that we're doing, plus the teacher training. So we're having those discussions with the provost right now, and we'll start launching into that in September. So thank you guys very much. Great job. Okay, so our last speaker for the morning session is William Panett. William is, I think, are you a junior at Worcester Polytech? Down in Worcester, Massachusetts, and he is working with Dr. John Stryker on an interesting project that's kind of growing another one of our core services for the university and the biotech community, and that's around drug discovery. He's going to be talking about some of the work he's done with the kappa opioid receptor.
Thank you for the introduction, Dr. Bilski. Like you said, my name is William Panett, and I've been working with my mentor, John Stryker, on a project we entitled Discovering Novel Kappa Opioid Receptor Recruited Proteins. So the kappa opioid receptor is a G protein-coupled receptor. It is found in many brain regions, including the striatum. It is associated with pain relief and stress-induced drug relapse. And this is our receptor right here. This is on the membrane. This is our cellular membrane. This is inside the cell. This is outside the cell. This is our protein. And this is our neurotransmitter that will bind to the receptor, and which will activate the protein. And I'll get a little more into this in a little bit. So 6' guanidinyltrindol, or 6' GNTI, that I will be saying for the rest of this presentation for my sake, activates the core. It is associated with anti-nociception. And it is important to note that it is biased against beta-arrestin 2 recruitment, unlike its standard core agonist, U69593. This is important because the side effects of beta-arrestin 2 recruitment has been associated with side effects such as aversion and dysphoria, and these are very harmful to people. And that is why 6' GNTI is an ideal drug, because it doesn't recruit beta-arrestin 2, which means that it could be possible for it to have anti-nociceptive properties without having the harmful side effects such as aversion and dysphoria. So this kind of comes back to an idea called functional selectivity, and why do drugs and compounds have different effects. These are our compounds or drug right here, and they're all different. And when they bind to the receptor, they actually activate proteins. These proteins then go out, find other proteins, and recruit them, which then get activated. And this continues in that manner, and it causes a protein cascade, which eventually creates a protein complex. These complexes are what cause the effects. So each protein, or each compound, causes different protein complexes, as you can see here. And these all have different effects, or have the potential to have different effects. So this kind of leads back to our hypothesis for this experiment. Because of its properties, 6' GNTI may recruit other proteins selectively when compared to the standard agonists, and that may provide alternative strategies for drug design. So here at UNE, we have an in vitro drug screening core headed by my mentor, John Stryker. They've been doing a bunch of stuff in vitro, including, this is in-cell westerns. These are confocal images of beta-arrestin 2 recruitment. And they are open currently to collaboration inside and outside of UNE. So the actual experiment I did, we tested 3 compounds, U69593, which is our standard core agonist, 6' GNTI, which was our functionally selective core agonist, and our vehicle was DMSO. And we analyzed 4 different brain regions for the frontal cortex, the brainstem, the periaqueductal gray, and the striatum. And you might ask, why did we choose those regions? Well, it turns out that those regions carry core kappa opioid receptors, and that's kind of important for a project that's focusing on kappa opioid receptors for them to have that. And we also chose those regions because we wanted to identify which region had the most effective activation. So we wanted to choose the region to continue analyzing further that activated the most effectively. And I'll get a little more into that later as well. So we used CD1 mice, and we had 6 mice in our sample. There were 3 vehicle and 3 U69 injections. Each mouse got 1 injection, but there were 3 mice per group, so 2, yeah. So we did intracerebroventricular injections, and this is our ventricle right here. It's a lateral ventricle. This is a human brain, not a mouse brain, but it's the same idea. The injections were 100 nanomole of drug in 5 microliter, and there was a 10-minute wait for the drug to circulate. And then the mice brains were dissected, and the brain regions were separated and put on liquid nitrogen in less than 15 minutes. And this is important because proteins decay quite quickly, and the quicker we can freeze them, the better chance that their receptors will still be intact when we analyze them. So we continue by doing a protein quantification because we needed to find the concentration of the brain tissues that we just dissected. And this is important for our Western blot because we needed to load equal amounts of protein into our wells for our Western blot. And the way a Western blot works is we have our proteins up here, and when we run the Western, the proteins will go down through the gel and get separated out by size. And then when you analyze those gels, thicker lines means more activation or more signaling of that protein. And so, for instance, this one has a lot of activation compared to this one, and these 2 aren't as activated as the others. The protein we analyzed in this experiment was ERK. ERK is a commonly found protein on the core cascade, protein cascade. And the 2 specific proteins were phosphorylated ERK, or pERK, and tERK, which is total ERK. Phosphorylated ERK basically means it's an activated protein, versus total ERK, which is just the presence of that protein. And this is the data we have. So this is our pERK, this is our tERK. Here's our quantified data. As you can see for this specific example, this is our frontal cortex. The vehicle is slightly higher than the U69, which wasn't expected. We expected the U69 to always be higher in activation rates. But this could be associated with the fact that the frontal cortex is quite variable in how much core it actually contains and how the activation occurs. And if you were going to look at our plots, you can see these 3 here are vehicle, these 3 here are your U69, and you can see that it's quite variable in which ones get activated and how much, which led to these results. This is our brainstem analysis, and right off the bat, you can see that U69 had a much more consistent impact, and it was to such a degree that we can confidently say that there was a difference between U69 and vehicle treatments. And you can also look at our Western blot over here and see that these three are definitely more activated or darker than the vehicle treatments. So this is our striatum. Although our U69 was a little bit more activated than our vehicle, we wouldn't say that anything for sure in this case, because you can see that they are very variable in how much they actually got activated. And this is our perioccipital gray region. This bar here is our error bar, and ideally, it would not be that big. We'd rather it be very close to this, because that means that the experiment was consistent and that all the values were done correctly. And then we look at our Western blot to see what happened, and you'll notice this line right here is very dark. That is probably why this occurred. And this was in our vehicle nonetheless, so that was very strange. And we looked at our loading as well to see if anything happened there. And you notice that all the loading was actually pretty consistent, and if anything, this vehicle was actually lower than the others. So this was a very strange value, so we decided we'd take it out just for the sake of analyzing the data without it. We got very different results. Our U69 was much greater activation than our vehicle, but we can't say for sure that this was actually a significant difference because of the fact that we took a value out. It actually lowered our sample size, and that's why our p-value actually came out to 0.08, and we were looking for a 0.05 p-value or lower. So in conclusion, it appears that there is some significant core activation in the brainstem as shown by the phosphorylated ERK. Although the PAG did show some signaling level, it was too variable to say for sure. So moving forward, we will analyze our brainstem to show core activation, and we will look at the functionally selected proteins that appeared into our protein complex. We will be using techniques such as immunoprecipitation and proteomics to analyze which proteins were activated by the U69 compound and the 6'-GNTI compound at the core. And if you were wondering what immunoprecipitation is, it's when we add an antibody to our tissue solution, and it will actually bind to our receptors. It's specifically designed to do that. And then these antibodies will bind to beads. These beads are very large, so when we centrifuge the solution, they will all go to the bottom. The proteins that are floating around up here can be easily removed, leaving just our antibodies, our receptors, and consequently our protein complexes that are attached to our receptors. And then we can analyze these receptors and protein complexes through a big machine that we call that method called proteomics. So for future directions, different functionally selected protein complexes will help in the creation of new strategies for drug design. If we know which proteins cause which effects, we will also be able to possibly create drugs that promote beneficial effects, such as anti-nociception, or minimize harmful side effects, such as dysphoria and aversion. This may result in new drug therapies in the future. So I would just like to thank my mentor and supervisor, John Stryker, Dr. Bilski and Uni for giving me the opportunity to work in their lab this summer, Lindsay St. Louis for providing important experimental procedures, and I would also like to thank Jordan Falloon and Henry Furland and several of the UNE interns for helping me prepare for this event, and the Center of Excellence in Neurosciences for funding my summer research project. Any questions?
I love making Ed run. That's really great. No. That was a very nice talk. I appreciate it. You mentioned a couple of things. One, there seems to be a lot of variability in those data that you presented. I was wondering how you would approach to address that. And secondly, I wish if you can talk about, you talked about a bias of some drugs relative to beta-arrestin. I'm assuming that means that some drugs turn on beta-arrestin?
Yes.
Which would alter your receptor availability. I wonder if you could talk about that relative to the ERK, Would you expect it to go up or go down?
Okay. So regarding the variability, well, in vivo studies, there's always a lot of variability involved versus in vitro because there's living things that can do things that we wouldn't imagine they would do. We could probably limit that by better lab techniques. I wasn't the most experienced person at first, but after I practiced a lot more, I got better. But obviously, I'm not going to be perfect every time in my brain dissections. So it would always just take more time for me to get better at those sorts of things. In regards to your other question, the beta-arrestin 2 recruitment, I guess I'm a little confused as to what you're asking there. Sorry. So you're saying...
Let's go back just to the variability first of all.
Okay.
I should have just asked you one question.
Yeah.
So you're talking about technical issues that certainly can cause variability, and there are some physiological issues that can alter or arise variability. Once you get to be proficient at it and we can dismiss the technical, what could you do about looking at and evaluating the physiological variability?
Well, we could do more behavioral studies because obviously, as you saw, we didn't really do any of those. We could see how the mice are reacting to those, so maybe it's a problem with how we're handling the mice in this specific context, so we could look into that. I can't really say any other experiments because I didn't really do any others.
Yeah, so maybe I'm not being fair. I was just thinking about increasing your sample size.
Oh yeah, definitely. That's another idea too. That I consider doing that. I'd like to do that, but obviously it's time and resources. You have to get a lot of mice, and these injections and experiments take a lot of time. And for one person, that's a little difficult. But obviously, ideally, I'd like to do that as well.
Okay, any other questions?
You're going to be nice to him, right?
Yeah. Hi, I'm Dr. Sheila Panetta. I'm an alumni of Unicom 2000, and I'm the director of the Maine CDC. And I'm also William's mother. First of all, I wanted to say, you all did such fantastic work today, very relevant to the needs of clinical medicine as an internist. So many things, the arthritis, we're working on that at the CDC right now as well. A big major problem, pain management, and also PTSD. Very relevant. So I certainly, as a clinician and the director of the Maine CDC, appreciate having such young people here today doing such fine work and moving forward. And as a graduate of this school, I know that Unicom provides a lot of bright, young osteopathic physicians that can go out and do things. As the director of the Maine CDC, I want to say for William, the research you're doing is so relevant because our addiction in this state is a major problem. We're the #1 prescribers in the United States of opiates. And we spend $44 million a year on people addicted to opiates. So my question for you is, what is the relevance of this in terms of the research you're doing in terms of preventing addiction long term to decrease cost?
Wait, are we talking about the addiction or the cost?
Both.
Okay. Alrighty. Well, like I said, these different proteins cause different effects. So if we can get more knowledge into which proteins cause which effects or which combinations of proteins cause different effects, we could probably figure out which proteins cause the addiction that she talked about and could possibly make it so people won't get addicted as easily to opioids and lower those harmful side effects. As for cost, I don't really know what the cost implication of this study was. I never really went into the marketing side of this. I imagine if it might be expensive, I can't really speak for sure on that.
Any other questions? Okay, well, thank you, William. And we're going to take a short break, coffee break out here, and then we'll reconvene at 10:30. Okay, welcome back. We're going to get started again for our second morning session. And our first speaker is James Havu, and he is going to be talking about the physical properties of iron-oxidizing bacteria and how they can be used for bioremediation. James?
Thank you, Dr. Bilski. My name is James Havu. I'm a student at the Florida Institute of Technology, but I live nearby, so I ended up coming home and doing research for the summer. My presentation will be different than a lot of the others because though I was working with bacteria, I'm a physicist. I know nearly nothing about biology. So there's going to be some physics and whatnot in here. I was studying a bacteria that you can see up here in the first slide. It's extremely common. It oxidizes iron. It can be seen in small ponds and ditches. It's just like an orange slime. And it happens everywhere. And it creates sheaths around it that have high iron and manganese concentrations. I'm going to talk in my talk. I'm going to talk about atomic force microscopy, which is the method I use to study these bacteria. The actual bacteria themselves, Leptothrix ochracea, and possible uses for them in industry, namely bioremediation. This is the device I used this summer. The one on the left it's an old atomic force microscope or scanning probe microscope. The one I used in the lab is nearly identical to that one. You put your sample up right at the top there. There's a set of lasers that detect the sample, and there's a scanner right there. One time we did go down to Woods Hole, Mass, and ended up using the Asylum Research Cypher, which was about 3 weeks old at that point. It's a truly incredible microscope. One of the images they took for us took 70 minutes to capture, and it would have taken us about 34 hours. So it's an incredible machine that we would like to write a grant to get a hold of, because it could really expedite the research process. The atomic force microscope works. It has a tip, and off the tip is kind of a diving board contraption with a tip on the end of that. And this is kind of the diving board with the small tip on the end. These tips are about 30 microns long, and the very end is just a couple atoms wide. And we use tapping mode, and basically what that is if you've got a sample, it literally goes over and just kind of feels the surface. And that's detected, and that's what our imaging comes from. And with this, it has a horizontal resolution of 10 nanometers per pixel, which is a higher resolution than the diffraction of light. So we actually see stuff that you can't see with an optical microscope because light does not work that well. There's 3 major types of images we used. These 3 are all exactly the same image. I took them at one time and put different filters on. The topographic one is literally just elevation. The brighter spots, the brighter spots are higher in elevation than the darker spots. The next one is a high-pass filter that measures sudden changes in height. You can see this spot right here is higher than the ones on the side, and there's kind of a line that shows that. The phase image I didn't really use that often. It's a very high contrast image, and what it really is useful for, and we never actually use this, is it determines how sticky or how hard the sample surface is. Because if it's hard, it'll actually get the tip out of phase for how it's tapping. The bacteria that I studied, the Leptothrix, it has 4 major stages of life. And this image right here is a really incredible one because it shows 3 of those 4 stages. In this image here, you've got this sheath right here. In the middle, there's these bright spots. These are actual cells. And around it is a sheath developing. It's getting iron put onto it. It's creating the sheath around it. This one right here is a fully developed sheath. And then this one here, the bacteria have left the sheath, and the sheath is now collapsed in itself. It's very old. This set of images has showed us that it showed how the process works through the development. And Another thing is the imagery, we can make 3D models, as you can see here, to really show that there is a good height difference in the way that they work. The reason these things use iron is they take an iron oxyhydroxide and oxidize it to make 2-line ferrohydrate. And in that process, it's a chemosynthetic process. And these bacteria use the iron to get the energy to live off of. What they specialize in is using iron. This is the 4 stages. This is actually a naked LO cell. And this is the protosheath being formed around it, a fully developed one, and then a collapsed one. You can kind of see up the middle where it's collapsed. It's kind of fallen in on itself, kind of just like this. This is the process we use to find where to sample. The 2 images on the left were taken a couple months ago by the person I worked with this summer, Catherine. And the image on the right I took with the atomic force microscope. And you can see through here, there's a small green line. We take the sample and stain it with a DNA stain called Cyto13 and put it under a fluorescence microscope and look to see where this DNA stain is. And then we'll look at it in a bright field and see what the bacteria sheets look like. And one thing that's so great about this image is you can see that there's definitely DNA right there. And over here, you can see where there's dark sheets like that, it's fully enclosed and you're not going to see anything interesting. But where it's a little bit lighter, the sheets have collapsed or are not fully developed, such as something like this where you can actually see the cells and the sheets at the same time. This is another image. This was taken this spring, I believe, before I was around. But it goes through the whole process of the DNA stain, the bright field image, and then this area right here in all 3 different types of images. Another thing that we can do with the machine is take cross sections. This is accurate to within 1 nanometer. We can do grain size analysis. Basically, there's fibers on the sheets. We can find out the size of those, of like the 2-line ferrohydrite fibers. The cipher that I was telling you about earlier, this image here took about 11 minutes to take. It would have taken us 5 hours and 24 minutes. And this image right here was the one that took about 70 minutes and would have taken us about 34 hours. So that's really impressive machine. It's beautiful imagery. You can really see what's going on. And one thing that was really cool was this fluorescence image I took a few days before we went down there. And you can see all the DNA, and they're able to overlay it so you can see where the cells are in the AFM image as well compared to the sheaths. And here's a little bit of the data we have. It's not terribly interesting, so I'm not going to spend much time on it. But one thing that was really cool about this was we determined at one point, we found a set of cells, and we thought they were LO cells. And we realized later on with this data that the cells were actually too large to fit inside of the sheaths. So we realized that of the 2 sets of cells we had, the ones that were prettier were not actually the ones we wanted to use. So we have determined which ones are the right cells, which ones are the wrong cells with this data. So something that hasn't really been studied at all with these cells is bioremediation. But it is one of the more likely possible ventures that could be done with them. Bioremediation is microorganisms breaking down pollutants. And one of the great things about bioremediation is that it's more or less natural. You're taking something that's natural and applying it to take care of something. These images, I think, are just pretty. This is the same area imaged at different times. And what my professor did is, this image was taken first, and you can see these large iron crystals. And he went through, and he started pressing harder with the tip, and realized that the crystals broke off. So these bacteria have, the sheaths have very brittle iron crystals on them, but it shows that they really do collect a lot of iron on the sheaths. It's a large source of it. These are some more images. This is all the same image 3 different times. And it again shows the large crystals, and I think that's just really pretty. Especially like that one just kind of popping out. Again, more crystals, a lot of iron that these things oxidize. So the question is, what could we do with this? If you were to introduce these into an area, a Superfund site, such as Katahdin Iron Works, or a lot of various old town dumps or something that have high iron deposits, this bacteria, once it's created the sheath, it literally takes the iron out of solution and oxidizes it, and becomes very easy to scoop up. You can literally just, use sort of a strainer and just pull it right out of the water. At that point, it could become a source of metal. Probably not a very good one, but nonetheless. But one of the possible things that we've looked into a small bit is genetic modification. If we could team up with a microbiologist, could it be possible to genetically modify these bacteria so that instead of iron and manganese, they deal with arsenic and lead? We actually found out about last week, there's a site out in California where they have found this specific species that is dealing with the iron and manganese, but also with the high arsenic concentrations. It has a high absorption capacity for arsenic. So that's one of the possibilities of what could be done with these. The question, where do we go from here? Later this summer, I was hoping to get some force modulation. Basically, you figure out the forces and the stretchiness, kind of the spring constant of the sheaths and the iron itself, to find out more about the physical makeup of the whole bacteria. So magnetic force microscopy could be done. That's what these images all are. Not stuff we took, but because there's iron, could it have a magnetic property? I don't know, there's a lot of places to go from here. We're not sure what. It's still a bacteria that's not very well known about. And as common as it is, it's not been studied very much. I'd like to thank Dr. Jamie Vizenka, who was a professor I worked for this summer. He's been working on this specific bacteria for a few years. He's been working with the AFM for about 20 years. Really knows what he's talking about. It was fun working with him. Catherine Maseko, she was my coworker this summer. She worked with Vizenka on a different project, and I worked with her a little bit, and she worked with me a little bit, and kept things interesting over the summer. The Uni Microscope Core Facility, which is where the microscope is, I had all the time I wanted at any time this summer to use that AFM. The Bigelow Lab, David Emerson has been studying these bacteria for about 20 years. We met with him. He had some great feedback. He's extremely knowledgeable in how these bacteria work. And Emily Fleming, who's a postdoc who worked with him as well. And Asylum Research, Jason Cleveland and Rob Kane especially, who let us use the microscope and did some imaging, and hopefully will get a little bit more back to us in a little while. And the Office of Research and Scholarship and Dr. Bilski, who funded me this summer. All right, any questions?
Hey, I've seen AFM images that image on the molecular scale. Do we have that capability?
What do you mean by molecular scale?
Like a molecule, like pentacene, I think it was.
The best I know about, yeah, here you, that's not a very good image. Yeah, stuff like this, these are just fibers. I'm not really sure how large they are molecularly. I know that Dr. Vizenka in the past has worked with DNA. They do have the ability, you can see DNA, you can resolve it. I'm not really sure how low molecularly what you're looking for, but this is about as good as you're going to get. And these tend to be about 60 to 100 nanometers wide.
Yeah.
I'm not sure where that goes to. But thank you.
Hi, thanks very much for that. What are the implications of this research on drinking water quality?
It hasn't been studied, but it's very possible that if these were introduced in an area with high iron or manganese, I have high manganese in my house, possibly they could remove the iron from the water, making it more pure. That would definitely be an area to study. I know, for example, these bacteria right here were found along the Kings Highway in Kennebunkport. My professor has some in his backyard. We're actually working on some right now they found in his backyard. So, I mean, they're very common. They do take iron out of solution. In areas with high iron, they probably could be used to purify the water. I'm not sure, but it would be definitely an area to look at.
Okay, thank you.
When you determine that the sheath material is what causes iron to crystallize around it, I'm wondering why one would need bacteria if you could just harvest the sheaths, let's say, to make like a filter, let's say, like French press coffee or something. Why not just have that sheath material in some way have water pass through it? And it's so much more complicated to grow bacteria, for instance.
Yeah. This way? No, it's the other direction. What they do is the bacteria have kind of a lipid sheath around them. And through the oxidation process, which I don't know a lot about, it's not my specialty, but they take the iron, they gather the energy from it, and at that point, they get it stuck onto the lipid surface. That's actually something that in all the research out there, we don't know how or why that is done. That's one of the things we're hoping to find out. And if we can find out how or why, maybe there is a way to do it without the bacteria. I don't know. I don't think anybody knows at this point, but we'd like to find out.
But that's a critical point, is that it's converting the iron into another form that then crystallizes on the substrate of the bacteria. So there's a two-step process there, where it's just straight filtration wouldn't give you the first part where it forms the crystals.
It changes the form of the iron, yeah. I want to say from iron 3 to iron 2 or vice versa.
Any more questions? Okay, well, thank you very much.
All right.
Next speaker is Rose Jacobson. She is working in the laboratory of Dr. Mike Berman, and she's going to talk about her own research in post-traumatic stress disorder and the connections between the amygdala and the hippocampus through the perirenal cortex.
Okay, so our research is on how the development of contextual fear in post-weanling conditioned rats is associated with enhanced activation of the amygdala and the perirenal cortex. So the goal of our research was to identify the contribution of the limbic system to the development of pediatric anxiety in rats. So we assessed both the environmental fear and fear to a particular stimulus, such as a tone. And our hypothesis was that the emergence of contextual fear conditioning depends upon the development of the perirenal cortex, which is the area of the brain that connects the hippocampus to the amygdala. And we did further studies that developed a paradigm by which contextual and fearful memory formation can be dissociated. So this is how we did our fear conditioning. On day 1, the rats were placed in a novel context they'd never seen before for 32 minutes, where they received tone shock pairings. On the second day, they're reintroduced to the same context to analyze whether they developed a fear to the context. And then on the third day, they were reintroduced to a novel context they hadn't seen before, where the tone is played, to see if they had formed a fear to the tone. And in adult rats, the tone shock pairings
create fear to both the context and the tone. So our goal was to see if at postnatal day 17 and 23, we'd have the same results. This is a little background on fear conditioning. It's a form of learning where an aversive stimulus, and we use an electrical shock, is paired with a neutral stimulus. We use odor, context, and tone. Previous studies have shown that fear to a tone emerges by postnatal day 17, and the ability to fear an environment emerges between postnatal day 17 and 24. The subjects we used in this experiment were the offspring of time-pregnant sprig dolly rats. This is the behavioral data that we collected with that fear conditioning paradigm. We used a 3 milliamp shock, and we saw a significant difference in the post-contextual freezing and not in the tone freezing for both groups. And then when we increased the shock to 1 milliamp, we saw a significant difference in both post-conditional freezing and tone freezing, which suggests that the fear system is developing during that crucial time. Then we did immunohistochemistry on the brains 1 hour after fear conditioning. We looked at CFOS expression, and CFOS is an immediate early gene transcription factor. Its expression is used to indicate neural activity because the gene is expressed when there's high neural activity, and it's highest 60 minutes after fear conditioning. And by examining the CFOS expression, we can discern which structures in the brain became active at postnatal day 23 and were not active at postnatal day 17. So the areas of the brain that we looked at were the perirhinal cortex, the hypothalamic nuclei, the hippocampus, and the amygdala nuclei. This is an example of a picture of the hypothalamus, and these are just examples of what the FOS cells look like when we count them. This is the results of our FOS data. As you can see, overall expression of CFOS is heightened in the 23-day-old rats, and in the 17-day-old rats, we see basically the same amount of CFOS expression in the basal nucleus of the amygdala and the perirhinal cortex. This suggests that the fear conditioning didn't activate these areas of the brain. And then in the postnatal day 23 group, we do see enhanced expression in the tone fear group for both these areas. This suggests that the fear conditioning did activate these areas of the brain. So because of these findings, it supports our hypothesis that the perirhinal cortex and the amygdala are undergoing development during that time period. And we used the hypothalamus as a positive control for our CFOS staining, and because we could see heightened expression in both ages, it suggested that our CFOS staining actually worked. So the next step for this experiment was to separate the context exposure from the shock. So we had half of our subjects were exposed to the context on day 1, and the other half remained in the transport cages where they had 5 minutes of context exposure. And then on day 2, all of the subjects received an immediate shock, and then on day 3 they were placed back in the same context to see if they could connect their context exposure from day 1 to their shock from day 2. When we did it with just an immediate shock, we saw no difference between the pre-exposed and not pre-exposed groups. So we added a varying time delay to day 2 so that when they're placed back in this context for their shock, they either have an immediate shock or they have a 5, 10, 20, or 30-second delay before they receive the shock. This is our data from that experiment. We did it with both 24-day-old and 17-day-old rats. So this graph shows that 24-day-old rats need between 10 and 20 seconds in order to differentiate the groups, because this is similar to what our data looked like originally, where in amongst litter mates, they didn't have any differentiation at 0 seconds, and there's really none at 5 either. So our next step would be to change our protocols so that they actually get this amount of time before they receive the shock on the second day. And then for the 17-day-old rats, we see virtually no difference between all the groups. This suggests that they can't remember the context that they were in, or they can't take both of those memories of the shock and the context and put them together. So to continue this study, we will add more subjects to both groups, and then we will also look at the CFOS expression in both the 17 and 24-day-old rats to see which neural substrates have changed over that critical time period to allow for the contextual and fearful memory development. That's it.
Very nice.
Thanks.
Do you think that delay that you see in the that you need to have in the day 24 animals,
Yep.
Between 10 and 20 seconds, do you think you could eliminate or shorten that delay considerably if you used also an odor?
We do use an odor.
Oh, you do?
Yes. We have 2 separate contexts. We have a square room and a round room.
Right.
One's black, one's white. We use ethanol and ammonia and yeah, so we do use an odor.
Okay, so you didn't mention that. Yeah, because previously I know you'd mentioned vanilla and strawberry, but
we don't use vanilla and strawberry.
yeah, but that's fine. That's,
yeah.
Yeah, yeah.
It takes between 5 and 7 seconds for hippocampus playcells to stabilize a new environment. So that's sort of the minimum it would take to recognize.
Yeah, even with an odor.
Even with an odor.
Yeah, yeah. Because I will then bet if you didn't have the odor, your delay would increase considerably.
Yeah.
And it's important that we only have one experimental room. So it's the same experimenter taking the same rats via the same cart into the same room. And so there's a lot of similarities even in our different contexts.
Yeah, that's very nice.
Any more questions?
Thank you very much.
Yep.
The next speaker is Haley Rutenberg. She is a summer intern. She goes to St. Anselm, and I believe you're from Kennebunk. And she's going to be talking about her work with Marine Plastics, which is a Biddeford-based company.
Thank you, Dr. Bilski. Like Dr. Bilski said, I'm Haley Rutenberg, and my summer project was to work with marine ecological habitats to take it from marine to murine, or laboratory-based products made especially for mice or rats. So just a little bit about marine ecological habitats. It's a plastic fabrication business, locally owned and run, situated right in Biddeford, Maine. Marine Plastics is actually a division of Marine Eco. It's distinguished for its ability to fabricate customized quality products. And the cool thing about Marine Plastics is it seeks to work with its customers to form the best possible product. So also a key part is the specialization and the customization of the products. And a couple products by Marine Eco: touch tanks for kids, the custom-built aquariums, and commercial lobster tanks, and also other marine plastic products, such as polycarbonate windows used in cars and boats. So the Touch Tank for Kids program is a 501c3 nonprofit organization actually created by the president, Joe Zucchero, the owner of Marine Eco, and it aims to educate children in the importance of conservation by giving them the opportunity to interact with different ecosystems in the classroom. And currently, Touch Tanks for Kids are found at more than 25 institutions across the United States. Another example of the products that Marine Eco designs are the aquariums. They are designed and fabricated per order according to the customer's idea of size and shape. And all aquariums assembled by Marine Eco feature particle filtration and environmental control systems that are easy to use and maintain. Marine Eco also produces a line of commercial lobster tanks that are also customizable based on the lobster business's requirements, as well as incorporating protein skimmers, UV sterilization, and carbon systems for purification purposes. The lobster tanks also feature an automatic drain down system, so this prevents lobster death in case of a power outage, so the water is actually taken from the tank so that the lobsters don't suffocate in their own waste. So the market for commercial lobster tanks is unfortunately limited. The lobster industry is not always reliable and is subject to fluctuations in both conditions of the lobsters hauled in, whether hard shell, soft shell, weather conditions, current changes, and as you can see, dockside prices. In the past few years, prices have actually declined due to aggressive competition between fishermen in the market. The market is flooded, or the dockside prices drop for retailers to maintain profit. So what this means for marine eco, taking into account the inconsistencies seen in the lobster business, into the purpose of the company, as well as their desires to broaden their market, Marine Eco decided it was time for a change. They wanted a challenge to enter into some other field of plastic fabrication that would allow them to grow and develop as a company. So as a business, Marine Eco has the staff, skills, and technology required to expand and branch out. The flexibility of the company in designing custom quality products, and also their willingness to work with their customers, sets them apart from all other companies. So the next step for Marine Eco, basically they need to pick a market of interest, find and solve a problem. So some of the advantages that they have, Marine Eco has worked with UNE in the past to design products for use in the lab, and these products are customizable. Other products typically sell stock product of certain dimensions and sizes, taking out the customization factor, and they're frequently costly. The reason that Marine Eco has this advantage to create such customizable products is that they use computer-aided design programs and CNC machinery for precision manufacturing. And this also saves time and money. Marine Eco is also important as a main business in the synergy that it creates in the local area. For example, they use Tigar and Wyndham to purchase CNC parts, or also CMC Tool and Die in Biddeford to help with rapid modifications of plastics and metals like thermobending. So these are products actually manufactured in collaboration with UNE. You've seen this is actually used in the osteoarthritic assay. This is a von Frey assay chamber. It tests nociception and sensitivity in mice. And this is an example of two different environments. There's actually the light and the dark, and the mouse is able, or rat is able to cross between these. So this summer, UNE actually approached Marine Eco with a new project. The problem was with the rodent restraint devices. They had limitations in the fixed diameter, the tail can slip free from the restraint device, and also the product can cause unnecessary stress for the lab animals. This is an example of one. So if the animal is too large or too small, the fit's not right for the animal restraint device, unnecessary stress for both the animals and for the researcher. So first off, we had to look at the competition. Kent Scientific Corporation, they sell tube rodent holders of fixed diameters. They have the optional nose cone feature, which is not shown here. They range in 6 different sizes that fit small mice to large rats, and the prices range from about $95 for the small to about $200 per unit for these. They also produce another product that actually shows the nose cone. The cone is adjustable, so adjustable for fit, and they have an open end at the nose for breathing. And the darkness of the cone helps the animal feel more secure. They have 7 different sizes available, and all are priced at $185 per unit. Harvard Apparatus features a flat bottom rodent restrainer with an adjustable tailgate, bottom slot for injections, 4 different sizes available, and the prices range from $129 to $140 per unit. One last example of the competition is the multiple tube type holder rodent restrainer. They're offered in 5 different sizes, and some have the inner tubes that can adjust diameter, but the prices range from about $368 to $608 per unit based on the size. So here is the Generation 1 and 2 Marine Eco prototype. As you can see, they have the opening in the front to allow the animal to breathe freely, and right now it shows the adjustable tailgate, and you can see the CAD design right here. The Generation 3, which they're currently working on, will solve the problems of diameter by including insertable sleeves that will adjust the diameter. They're also looking to tint the acrylic to reduce animal stress, as the darker colors help put the animal at ease. And also, they will be incorporating the removable head cones into the Generation 3 tube. Here's the budget proposal actually for the Generation 3 tube. Based on the competition, The typical animal restraint device is about $240. The Generation 2 tube, designed for rats of larger size, is estimated to be about $100. The Generation 3 budget right here would estimate that the cost would be about $181. So it's about an $80 difference. So the next step is actually getting started. One of the opportunities that we've been looking at is the Maine Technology Institute Seed Grant, which is offered 3 times a year to Maine businesses, and it supports early stage research and development of new products. And ultimately, the goal of the MTI Seed Grant is to help Maine businesses expand and develop, and in turn also aims to create more jobs and increase revenue in the state. So this summer I actually am working with Marine Eco and UNE, and I'm drafting the grant application. So UNE and Marine Eco will be working together to design and test the prototype. And some of the advantages that Marine Eco has in terms of this grant: they're already skilled and experienced when it comes to producing plastic products, and no purchase of tech or machinery is needed. They're already a fully equipped plastic fabrication lab. So the goal of going and attending conferences is to show the life sciences market what Marine Eco can do and what makes them special and sets them apart from all the other companies. Some of the upcoming conferences include the Experimental Bio Conference, International Bio Convention, and the 2013 AALAS Conference. So in order to promote these products, Marine Eco hopes to attend conferences and conventions to advertise to potential customers. The costs and requirements of attending these have to be taken into account, however. The MTI Seed Grant does not fund marketing and advertisement, so this would have to come out of the company, but this is a good opportunity to expand. For example, the International Bio Convention is about a $400 minimum of a 10 by 10 square foot booth. And the AALAS conference is about $1,700 to $1,900 per booth space. So it's a good opportunity, costly, but in the end, it's a good risk to take. So the future for Marine Eco, basically to develop new innovations within the life sciences market, find a problem, find a solution. Display prototypes at trade shows and conferences in order to spread awareness about the company. Develop a network of customers within the scientific community. A good example of that is the partnership with UNE. And growing businesses help Maine. So as Marine Eco expands and grows in the life sciences field, the local community can in turn benefit from the creation and retention of jobs. And as a state, Maine has more to offer in the fields of science and technology. So thank you very much for this opportunity. I'd like to thank all of the professors who lectured us this summer, came in, shared what they knew, all the sponsors of the program. Thank you so much. Without you, this would not have been possible. Sam from Marine Eco, thank you so much for answering my questions. Thousands of questions, lots of emails. Jordan and Henry for running the program, organizing everything. Thank you so much. I don't know where Henry is. There he is. And also the Bilski Lab, Denise, Jim, Amanda, and Lindsay for welcoming us and showing us the ropes. So thank you very much. Now open for questions.
Nice job.
Thank you.
How involved do you think UNE will have to be for this company to be successful? Do they have the ability to come up with the problems and solutions on their own, or is this going to be long?
Yes. UNE is giving them basically a foothold into the life sciences community, but Marine Eco definitely has the expertise and the background in order to design new products based on problems. It's kind of what they do as a company, and I believe that they can successfully bring it into the life sciences market.
I'll just speak loudly. Where do you see the market for products that you could make being 10 years from now? In other words, would there be less reliance on whole animals, more molecular work? What would that imply?
Yes.
For the things that would have still unmet needs, but that might be addressing different particular circumstances.
Well, eventually technology changes and equipment needed in the lab grows outdated and you need to replace it. And Marine Ecological Habitats has the ability as a plastic fabrication company with the computer-aided design programs and the CNC machinery, I believe, to be able to meet those needs to to identify and be able to fix the problem. So.
But going for free associating, if people have pets and they like pets, is there a veterinary application, let's say, to administer injections in a veterinarian's office? I just am making this up, it may be a dumb idea, but other markets that you could use your fundamental skills and capabilities for, rather than focusing on today's unmet need.
Yes, definitely. I think there is a great opportunity for that. This is first designing products for this is just a kind of a way to get out into the public eye in order to spread awareness about what the company does. And I'm sure as soon as Marine Ecological Habitats has like the opportunity to expand into these different realms of like veterinary or other animal care, I'm sure that it would be a good opportunity. So, if that answered your question.
Thanks, that was great. I have a question thinking about Maine and different companies. Maine, Jackson Labs comes to mind. I'm wondering if this company has made any connections with Jackson Labs in terms of what they may need that the company might be able to provide.
There's been a preliminary meeting between the two. As of yet, I don't think anything solid has been discussed, but that is definitely an opportunity that we're going to be looking into.
And how, I guess, I'm also just thinking in terms of process, how does that happen? Like who, if the company, they say, I have this, I can fill this niche here and we can make these products custom built, and there are people here at our university who have contacted them and say, hey, can you help us out with these things? And they want to expand, and then here's Jackson Labs. How do they know who to go to and who to talk to about that? I guess that's something that I would just wonder about.
I think it's all about connections. All the work that they've done for UNE, for example, would be a good foothold in order to get to the companies. So it's really just about showing what you've done and having the testimony, so to say, to back it up, to be able to prove what they've done, prove the efficacy of all the products that they've made, stuff like that, I think.
I'm just going to take a little sidebar here. Karen Gregory's on our board of trustees, and she's also heading the Maine Bioscience Group. And maybe you can comment on the connectivity.
Sure, thanks, Ed. I think that's one of the benefits of any company and any organization in Maine joining the Bioscience Association, because that's part of what we try to do, is to help companies showcase what they do and what they can do, and to try to make those connections to the academic centers and to research institutes. So to the extent that Maine ECO has not yet been involved with BAM, they really should consider participating. Mm-hmm. I think in general, a company like that certainly needs to find the science folks who are actually finding the problems in the labs particularly, and they're the ones that can then drive potentially, to the purchasing folks. But we're certainly making a big effort. At BAM, we just launched a new website. It's going to have a lot of opportunities for members to post things and for them to be at conferences like our fall conference to be able to showcase what they're doing. So I urge them to consider talking with me further. Thanks.
Thank you. That's a great opportunity.
And the University of Maine system is also working on a program that is teaching innovation and entrepreneurship. So it is starting to expand, and we think linking these groups with people like yourself to help. So thank you very much, and we'll go on to the next speaker. Already seen the diversity of the types of presentations today, from a very basic atomic force microscopy looking at the physics or the physical properties of these bacteria, to the translational types of research being conducted in the biomedical field here, and also the connections with the companies. Brittany is a graduate of our university as an undergrad. I think, were you the first class of neuroscience majors?
Yes, there were 8 of us.
8 of us. So she was a pioneer there, and then she went to our osteopathic medical school. And during her first year, she became connected more with the outreach program, wanted to continue to expand her horizons in research. She had conducted basic science research with Mike Berman, and looking for clinical opportunities. And we had this affiliation with Maine Medical Center, and David Cedar and Rich Riker in the neurocritical care unit. And it was a perfect fit. You got interviewed and got selected to represent us for this year. She's going to be talking about her clinical research project. Brittany.
Thank you. Hello, everyone. As Dr. Bilski said, I was able to work in a clinical realm looking at the associations of early hemodynamic factors and oxygenation with outcome in cardiac arrest survivors treated with therapeutic hypothermia. So every year in the United States, about 300,000 people will suffer a cardiac arrest, and 40 to 60% of those people will experience return of spontaneous circulation, or ROSC. Their heart will start beating again after resuscitation efforts. Prior to the age of therapeutic hypothermia, about 20% of those patients would actually survive to hospital discharge. Today, with therapeutic hypothermia as a standard of care for comatose patients after cardiac arrest, we've increased that survival rate to 40% approximately. This is great. It sounds like we've made obviously great breakthroughs. We've doubled our survival rate. But the clinical problem that we still face is that we still have to tell 60% of families that their loved one isn't going to make it. And this has prompted investigations into what variables influence that neurologic outcome. Kilgannon and his colleagues have published several papers in this field. One recently has shown an association of early arterial hypotension associated with increased in-hospital mortality. This is rather intuitive. Hypotension would also mean hypoperfusion to the vital organs, which in turn would increase your risk of mortality. The same paper showed an association between hypertension and a better outcome. Again, rather intuitive. A second paper published by Kilgannon and his colleagues showed an association between arterial hyperoxia and in-hospital mortality. This one's not quite as intuitive. One would think that someone who suffered an anoxic event such as cardiac arrest would benefit from being given as much oxygen as possible. But his paper showed that a PaO2 level of 300 or greater was actually linked to detrimental outcomes. And the hypothesis right now for the reasoning behind that is that this increased oxygen level is also increasing reactive oxygen species in the brain, which leads to lipid peroxidation of the neuronal membranes and actually causes secondary neuronal damage. It is important to note that these published papers are associations. They're not cause and effect, and therefore they're considered a low-grade evidence. They were also done on populations that were not treated with therapeutic hypothermia. So what we want to know is what are the real target ranges for blood pressure and for oxygenation in order to prevent secondary neurologic injury in cardiac arrest patients who are treated with therapeutic hypothermia. We're particularly looking at blood pressure and oxygenation because they're really easy to manipulate by drugs and simply placing an oxygen mask on or off a patient. Our generalized hypothesis is that manipulation of these hemodynamic and biochemical factors can mitigate the risk of secondary neurologic injury in cardiac arrest patients treated with therapeutic hypothermia. And the specific aims of this study were to look at the occurrence and frequency of hypotension, hypertension, and hyperoxia in cardiac arrest patients treated with TH, to determine whether there's a relationship between early arterial hypotension and the neurologic outcome of these patients, and to determine if there's a relationship between early arterial hyperoxia and the neurologic outcome in these patients. Our patient set was taken from the Maine Medical Center database of cardiac arrest patients treated with therapeutic hypothermia between the years of 2005 and May of 2012. There was a total of 320 patients, and we then excluded those who were under 18 years of age, those who died within 24 hours of ROSC, and those with an unknown ROSC time, as they also had an unknown downtime. Our final sample size was 265 subjects. Of those subjects, we first looked at demographics and cardiac arrest information, including gender, the initial EKG rhythm, whether it was a ventricular arrhythmia, PEA, or asystole, whether the arrest was witnessed, whether the patient received bystander CPR, whether the patient suffered a STEMI or ST elevation MI, and whether the patient was in shock on arrival to the ICU. Then we went on to look at the data pertaining to our specific interests, starting with blood pressure. We looked at both systolic blood pressure and mean arterial pressure because we don't know yet which one is a better prognosticator. With systolic blood pressure, we asked two questions. did the patient experience 2 or more episodes of a systolic blood pressure less than 100 in the first 6 hours? And that's what we termed as early hypotension. We then looked and said, did they experience 2 or more episodes of systolic blood pressure greater than 160? And we termed that early hypertension. We did very similar for the mean arterial pressure, adjusting those values to less than 60 as our hypotensive group and greater than 100 as our hypertensive group. When we looked at the oxygenation levels, we first looked at the initial oxygenation level taken on admission to the hospital. This was usually very early. It was usually done in the emergency room, and in all patients, it was within 4 hours of the time of ROSC. And patients were classified in one of 3 groups. They were either hypoxic, if their PaO2 is less than 60, normoxic, if between 60 and 299, and hyperoxic, if they were greater than 299. We then looked at the PaO2 levels over 24 hours, and we assessed whether each patient's highest level was greater than or less than 300, and whether their lowest level was greater than or less than 60. And what this told us is, was the patient ever hyperoxic or hyperoxygenated during the first 24 hours, and whether they were ever hypoxic during those 24 hours. As continuous variables, we looked at age, time to ROSC, or the downtime before their heart began beating again, BIS1, which is an electrographic way of measuring neuronal activity, peak neuron-specific enolase, which is a protein biomarker for neuronal damage. We looked at the highest level over 72 hours after ROSC, and the number of vasopressors each person was on in the first 24 hours after ROSC. Our outcome measurement was hospital discharge cerebral performance category. This is a 1 to 5 scale. It's been standardized and generally accepted as the way to assess neurologic outcome in this patient set. A score of 1 to 2, which is mild to moderate cerebral disability, is considered a good outcome, and a score of 3 to 5, which is severe cerebral disability, coma, or death, is considered a poor outcome. For our statistics, our bivariate model looked at continuous variables as mean and standard deviation, categorized or dichotomous variables as the number and percentage. We performed t-testing chi-square, and a p-value of less than or equal to 0.05 is considered significant. We also did a multivariable model looking at other variables that are known to influence outcome with the hopes of determining whether anything that we're interested in is an actual independent predictor of outcome. And in that we included age, rhythm, time to ROSC, whether the arrest was witnessed, whether there was bystander CPR, and then our variables of interest. So results. First we looked at the demographics and cardiac arrest characteristics. You have your good outcome group, poor outcome group, p-value, and degrees of freedom here, variables around here. There were significantly more patients with a ventricular arrhythmia who had a good outcome than a poor outcome, their p-value here. There was a significantly shorter downtime for good outcome patients, and significantly more witnessed arrest and bystander CPR for good outcome patients over poor outcome patients. Looking at the clinical characteristics, patients with a good outcome had a significantly lower peak neuron-specific enolase level. There were significantly more of them who suffered a STEMI, and they had a significantly higher BIS1 value than those with a poor outcome. This makes sense and has already been published and was what we expected, as the higher BIS level indicates a higher level of neuronal activity at the initiation of therapeutic hypothermia, which would indicate that there was less, or hopefully less, neuronal damage incurred during the cardiac arrest event. When we look at our hemodynamics or blood pressure data, we have the systolic blood pressure data on top here. There were slightly more patients who were hypotensive in the poor outcome category than the good outcome category, and slightly more hypertensive patients with a good outcome than a poor outcome, but clearly not strong enough to be statistically significant. The trend's a little stronger when we look at mean arterial pressure. Again, of the hypotensive patients, there were more that had a poor outcome than a good outcome. And again, in hypertension, more good outcome than poor. Getting closer, but still not statistically significant, but it is trending in the same direction as the previously published works and where we would expect it to go. Oxygenation data wasn't quite so clean. First of all, there were very few hypoxic patients on admission to the hospital, which could be good. That's telling us that hopefully our emergency medical services are preventing hypoxia as much as possible. But unfortunately, there's so few that it's probably not statistically reliable. When we look at the normoxic patients on admission, there were actually slightly more in the poor outcome than good outcome categories. And when we look at hyperoxic patients on admission, there were slightly more with a good outcome than a poor outcome. Those are both flip-flopped from where we would expect them to go, but it is important to note again that they're not statistically significant. Our multivariable model gets a lot more complicated, looking at odds ratio for the unadjusted and odds ratio for the adjusted models. First off, I will mention all of our confidence intervals cross over the value of 1, so none of them are statistically significant, but you do see another trend. Prior to adjustment, the odds of a poor outcome is greater when hypotensive than when hypertensive. And after adjusting for other variables that influence outcome, the trend remains but is much smaller. So why are our results so different from what's been published? The first thing is sample size. Ours is 265. The published works is 6,326. Ours is a single center study, and theirs included 11 centers. This is probably the biggest problem in our blood pressure data, where we were trending in the right direction and trending towards significance, but not quite there. Data interpretation was also a big thing. We categorized our data. That's sort of contraindicated with what we're looking at. We're trying to look for ranges, and we kind of guessed where those ranges should be, especially with our oxygenation data. We classified patients as hypoxic, normoxic, and hyperoxic, which severely decreases the value of our data, and also places patients, for example, with a 299 oxygenation level into normoxia and 300 into hyperoxia, where clinically those patients are really very similar and probably at an equal risk of oxygen tension damage. So that is another option. Our subjective outcome assessment, the CPC, although it's been used in many published studies and it's considered the accepted way of assessing outcome, it's really subjective. It doesn't take into account withdrawal of life support. Any patient who died in the hospital went down as a level 5, a death, and a poor outcome. However, if families withdraw life support before we've fully treated, there's no way to know if those patients would have been a good outcome. It also doesn't take into consideration secondary cause of death. Some patients wake up in the ICU, they can obey commands, they're, for all intents and purposes, neurologically intact, and our treatment has worked, but they die in the hospital for another reason. They get a secondary infection, or they have another heart attack, and they're still considered an in-hospital death with this outcome measurement, and really were a good outcome before that secondary event. And perhaps the most obvious, the published data was not on patients treated with therapeutic hypothermia, and ours were. And there is the possibility that therapeutic hypothermia provides a protective mechanism against oxygen damage and even against fluctuations in blood pressure. So what now? We want to reanalyze the data using those continuous variables, especially for our oxygenation variables. We also want to reanalyze using more objective measures that are not influenced by end-of-life decisions, such as the peak neuron-specific enolase, MRIs, or the BIS-1 score. We'd also consider including comorbidity data into the multivariable analysis because, as you may expect, the intensity of illness is also a confounder for outcome. And we'd like to consider expanding our sample size, including other institutions, or potentially, once we've worked out these other kinks, using a prospective approach with patients as we go along with treatments. So before I take questions, I'd just like to thank Dr. Bilski and the Center for Excellence in Neuroscience for having me, and especially the Michael T. Goulet Foundation for sponsoring my research. It was an awesome opportunity, and I'm very grateful. So, thank you.
So that was incredibly thorough. Thank you. I had a few questions and then could cross them off as you went. But I was curious if you separated patients that were brought in from a pre-hospital setting in your demographics, and whether you looked at trauma versus medical codes and kind of broke those apart.
We did not. We do look at where the patient goes after hospital discharge, but we did not separate based on those who experienced their code, for example, in a nursing home where maybe care would have been more immediate than someone who was just on the side of the street. That is sort of why we plugged in bystander CPR. Although we don't consider hospital or nursing home faculty as bystander. As far as your second question, we do not separate traumatic cardiac arrest versus medically oriented cardiac arrest. And that is another difference with the published works. Most published works are non-traumatic cardiac arrest patients. Our sample size is so small as it is that we've included both.
Any other questions for Brittany? Okay, well, oh, one more.
If I recall your table, the BIS monitoring was predictive of outcome.
Yes, it is.
And
And that's another publication that we're actually working on because it's a rather new.
And that would be logical because BIS, at least in theory, could tell you the potential of the nervous system to generate a complex signal going in the direction of being conscious.
Yes.
So it's an index early on of very useful, very significant prognostic value. In the area of monitoring of patients under general anesthesia in the OR, increasingly there are devices that can directly measure cerebral oxygenation and perfusion in a non-invasive way. They may use some infrared or other probe. Have you found that those are becoming more widely applied during therapeutic hypothermia? And if so, could that be another encouraging thing to monitor, like parallel concept to the BIS, although
Yes.
different technology?
That's actually another direction that we've already started to head. Dr. Cedar, who was the PI on this project, is working. He's already started with an implantation of an interventricular device that measures cerebral perfusion and oxygenation. And they've just started to get IRB approval to start using that in therapeutic hypothermia patients. It's already being used at the hospital in the neurocritical care for other diagnostic purposes. So it is a direction that we're heading. It's far behind the BIS data for our purposes, just in terms of getting to the point where it can actually be used as a prognosticator. But the BIS data is actually in the works. They're also looking at the BIS after 6 hours from ROSC, because right now it's very inconsistent when it's being applied to patients. By standardizing it to looking at a value that's 6 hours after return of circulation, we can even get a better grasp on how good of a prognosticator it could be. So they're very, very much working on that, and even EEG data right now.
Okay, well, thank you very much. The last student presentation is Michael Stanley. I got to meet Michael through his dad. I was giving a talk at a Kiwanis club, and his dad came up to me and said, you needed to meet this guy that I know, his son. And he connected us through email. Michael is attending Harvard University and has deep ties to Maine, and I was impressed in his emails about his commitment to coming back to Maine and wanting to do things around economic development. So I thought he was a natural for the summer intern program. And I gave him perhaps one of the most challenging of the projects, and that is to tie in all of the business community, primarily focused in southern Maine, around the biotechnology that's going on. And I think it extends from Sanford down to Kennebunk, up in through Biddeford, Portland, up to Westbrook, and beyond that. And I think part of the presentation is going to be talking about that. Michael's also gone above and beyond by inviting some people that he knows through the business community and local politicians, and I appreciate them spending the time here too, learning more about the university and the program. So without further ado, I introduce Mike Stanley.
Thank you. Yeah, so thank you very much today for bearing with me. I am the last one, hopefully one of the least to say. I think I actually have a lot, but I don't think it might be very good. So today we're going to talk about Maine, and we're going to talk about something particular, that Maine wants to advance an innovation economy. And our biotechnology clusters are key to that. Biotech, of all the technology clusters, I think here in UNE has the greatest potential for advancement. So very quickly, I'm going to review some of the state's interventions, and then we're going to look at what the landscape looks like so far, and then at the end I'll make some recommendations. Hopefully I'll be able to get to the end. So what do I mean by a tech cluster? So industrial clusters are defined by the products that they make. So we're talking about a technical cluster. It's by its skill set. A good example of this would be what Haley was talking about. A company that knows plastics and knows how to bend them and work with plastics very well can make one product, yes, for lobsters, but it can also make other products, in this case for mice. Another example would be what Alex talked about today. We had a company which was familiar with one type of gel technology. I believe it was for sauces, right? Is that correct? And now sauces for mankind is food for mousekind. So it's a skill set. It's not the product. In Maine, we've decided to say there are 7 tech clusters. Biotech, of course, you see touches quite a few others, and we can use science and innovation in one cluster and leverage it to benefit another. And so we can, rising tide lifts all boats, sort of. As you can see, another thing I'd like to highlight is that there are different levels of the clusters. Some are sustainable. There are more historical ones, like forest products. In agriculture, we know them very well, and we've grown up with them. Others are new, and so they're just starting out. So what is the life cycle of a cluster? How does it work? So you have to start with a knowledge center. An institution like UNE is a great example of that. And then there's networks, and there's knowledge spillovers. Something comes out of that that's commercializable, and a guy goes, I got a great idea, and somebody else says, I want to buy it. So you have entrepreneurship, and then hopefully a product comes out or some form of distribution. I'm going to use as an example what's been going on with the Maine Composites Alliance and its composites incubator at Brunswick Naval Air Station, so very briefly to outline this. So SMCC in UMaine provided students and knowledge. A private consulting company provided knowledge and skill sets. And Fairchild Semiconductor donated equipment. This all made its way over into an incubator where students are taught, and the lessons that they learn advance the science. In this case, they were looking at materials testing. How do we advance the particular composite materials that we want to look at? And then, as well as styrene substitutes. So with some entrepreneurship, they produce a product that is relevant to the industry itself. So it's self-sustaining here. And this is what drives the train. In biotech, for example, we'll talk about later, UNE right now is in a great sort of consulting capacity. This internship program is a good example of it. Another would be what John Stryker is doing with the Discovery Corps, screening drugs. It's in a capacity right now that it is engaging and interfacing with industry, but how do we corral all that energy and all that talent into what could be an incubator? I think we can do it. So let's look at the past efforts. Since the mid-1990s, starting with a $20 million bond and being renewed periodically up until just about recently, we were investing in R&D. We were investing in research. Macroscopically, we did great. We met capacity. We went from 49th in the country to 35th in terms of research. For every $1 that we put in as taxpayers, we got $12 out, which is about on par with the amount of money that we were putting in for that. So research and development, we were doing that. And if you total everything all together, you get $524 million. So very good investment, and we grew our research capacity now into something that actually could be considered knowledge centers, something which really can drive it. But that's a lot of R, not a lot of D. That's partially how things were set up, but also because development is a very nebulous thing, very hard to just say go and produce knowledge, but the knowledge generators aren't always exactly the same people which do the development. The old paradigm from Paula Stephen, great book by her, and I can send you that, is that research is the realm of academe, whereas development is industry. And we're going to challenge that paradigm. I think what you've seen today is a challenge of that paradigm, with students learning how to advance and develop industry using skills that were in research here at the university. So let's look at the public players. They received over time $70 to $120 million, and they used that money appropriately, as you saw, 1 to 12. Very well done. But if you look at things, it was a very, if you build it, they will come. You know, if we give the money, research will be produced, and then from there, development will come. Where really, as you saw in the cycle, what drove the train was the translation of it, was the guys which could look and say, well, what is commercializable here that you're giving me? And then how do I find the market for that? So as far as commercialization, it wasn't proportional with the amount of money that we were giving. If you look at STEM education, is an indirect thing. Well, maybe we got more in terms of STEM students. That is not correct. About the same. We didn't lose any, but we didn't increase significantly based on the amount of money that we were putting in. And if you look at graduate programs or graduate students that were being produced, 2 to 3 times below national average. Now, we've got a new graduate school which has come into being, so those numbers will change. But there's some catch-up that needed to be done, and we're doing that now. Privately, in terms of state funds for industrial R&D, that was reduced by about $100 million. But you did see, proportionally, they were producing 30%, if you look at it. So 253 businesses, 30% of those filed patents or intended to file based on research that was funded through the state. So they were producing, and it was on par with the numbers. But their big concerns were, we need skilled labor, we need people which actually know the science and can actually work with the science, and we needed more capital. One of the significant problems we have in Maine is venture capital or some form of funding. It's very hard. A lot of it's down in Boston. A lot of Boston doesn't like to come up or even know that we're here in some cases. And I can tell you an anecdote afterwards about that. So how do we pair this? How do we match the two things together? That's the landscape that was written out in the Maine Tech Cluster recommendations. So you see universities. You see pharma back and forth. They have an idea. They need some help. That goes back and forth. Biotech is the big thing. This is something which could be our networks, our knowledge spillovers. This could be our entrepreneurship. This red line, which is broken, I want to make a solid line. And I think UNE, through its collaborations already, and with further collaborations, will make that black line. So knowledge, networks, and then products come, product distribution even to pharma. Oh, we can look very briefly. Some people say, well, they're high-tech jobs, or they're this, or they don't pay. Well, they do pay, actually. There's 3,700 people that are biotech employed, and about $47,000 for average wages. So they're good-paying jobs, and they are here. UNE is a model as an institutional transfer pipeline. It's fantastic if you look at them all. It has a patent. It has patent officers. It has a licensing budget. It has well-established protocol and procedures. It knows what it's doing, and it can facilitate the pipeline. So this is a key partner with little companies that want to produce something. If you look at startups, Orono produced the most. This was a study since '98. UNE had 2, ICO being one of them. It wasn't, although one UNE was surveyed, this didn't come up in the particular R&D recommendations list, which was odd. But one thing I do want to point out is Jackson Labs, which only produced 1 startup. So Jackson Labs, which is, they produce more publications than any other institution in the state. Longest here, but only 1 startup, because translational capacity is what's at issue. You have to know about the pipeline and how to generate it. Jackson Labs is very disciplined, and they know what they need to do. They're interested in genomics and genetics, and their product is mice, and they advance through that. So they're very specialized. Translation doesn't have to be specialized, and so that's something to keep in mind. Key events to all these startups when you interviewed them. They all said, we need an institution to house us, to help us, to foster us, and funding is always good. As you know, it takes a little money to make money. So MTI has been there, and we're major resources in SBIR grants. If you look at entrepreneurship, saying, well, what if we do all of this, but nobody in the state actually wants to do this? Nobody has the ability to go out and start a business. Well, that's not true. We're 7th in the country in terms of entrepreneurial intensity. And a lot of that has to do with Mainers are self-employed, 3rd in the country for self-employment rates. So we're primed for entrepreneurship. A significant problem was the bottleneck issue. Companies are good. They have a product. They think they're okay. They're a little risk adverse, as everybody can be risk adverse at times. So an institution which can help them through those troubled waters and help them negotiate issues that they're unfamiliar with, that's the key to eliminating that bottleneck. And then those difficulties we'll talk about right now. If you looked at all the recommendations, there were several reports beginning in 2006 and then 2008, and then there's another one coming out in 2012. And you boiled all of them down. What you ended up with was, I have issues with human capital, translational capacity, and networks. Don't have hands, don't have minds, don't have a place to sell the product, don't know who anybody is. So if we solve those problems, we can leverage the capacity that places like UNE have to increasing our innovation economy. And if you look at some, so I've outlined in black things that we are doing, in red things that we might do if you took me seriously and believed me, which sometimes I don't. But K through 12, outreach programs. You've heard Evan and Trevor talk today, and hopefully I've been very impressed, as I have, about what they're doing in class, and some suggestions were made what to do after class and online. In the state, there is a program now called REACH. So REACH is an example of meeting some of these issues. But our outreach program at UNE is a key to the in-class. And being one of the first there, one thing is to not be redundant. So if there are people which can help, don't help by starting a new program, join ours and figure out how we can spread that. Because if you have multiple, left hand doesn't know what the right hand is doing. Another serious thing is the STEM diploma. When you graduate from high school, as Falmouth, I believe, does. Yes, I'm getting a nod from the rep, which says they do. Falmouth has a STEM diploma. I graduated from this high school. I've taken so many STEM classes. That's something other schools could adopt, which would help put us forward in saying STEM is important in the state of Maine. And then, of course, aligning standards to industrial needs. This is something which is very difficult, but one great example is what IBM has been doing in New York. So it went into schools which doesn't have a vocational program. And they said, well, we need, as IBM, we need people to do work for us. So let's train them. We'll train them in high school. We'll make our own voc program, essentially. And when they graduate from high school and they come work. So they have a job when they leave. Maine's tough in that we don't have an IBM. We don't have big, but we have enough small little guys in related issues, as you saw in the clusters, that can leverage in the same way. And then higher education, without a doubt, from everything I've looked at, this program is the greatest example of being able to advance clusters in technology, particularly biotech, at a higher educational level. Access aid program. So some schools have faculty aid. So if a lab doesn't have enough money to cover, we'll say, an intern, the university will pitch in half and the lab itself pitches in half. One way you could look at this is to do this with businesses that are within the clusters. And to be able to say, all right, you may not be ready for an intern to pay yourself because you're relatively small, but maybe for some way we can share an intern. Or MTI, for example, or institutions like that that are looking to advance can look at this as an opportunity. And then make sure you keep entrepreneurship in STEM. So much academia has emphasized, a lot of people don't remember that industry is a part of that. Just as academia learns what's next big thing from industry, industry needs to know what's next from academia. And then, as Ed mentioned, foster Center for Student Innovation. We have student innovation competitions. Having representatives and people from government to come and listen to students and have students to come and listen to them is a significant portion. Listservs, as we've just found out, are an incredible way for students to figure out what's going on in other parts of the state. And then for institution to industry, things like what UNE has done with the Center of Excellence. To take all the people that can solve problems in a particular area, put them together, have them talk to each other, look at a problem and solve it. As I mentioned with the Drug Discovery Corps, or in helping to test new products for different companies. Incubators we've mentioned, and then UMaine and patent, I think, is something to emphasize, their patent program. And then networking. One thing I thought before we leave, and I think I still have some time left, is for tech talks. If you could have your trade representative, your university person who's familiar with the science, and then a business sit down, look through the recommendations that were made. There was a technology action plan, for example, and see what can we actually do to make this from a recommendation to an action in a year, or in 2 years, or in 3 years. Sit down and talk with each other. And then, of course, innovation zoning. Being able to say related industries, like a biotech to an agriculture will say, to work together in an incubator setting to solve problems that require more than just one industry. And so I'd like to thank you. Again, everybody who has been thanked already, I thank them. There's far too many to name, and so I won't name all of them, but Dr. Bilski, Jordan and Henry for facilitation of the program, all the interns, which was great to bounce ideas off of, and then, of course, our sponsors, without which this program wouldn't be here. So thank you, and I'll entertain any questions now. Or we're just hungry.
All right, I'll hold you up from lunch.
Cool.
Just a quick one. You had said that you wanted to make the dotted red line
Yes.
from biotech to more of a solid line.
Yep. Do I have any ideas?
Do you have any direct ideas?
Well,
on,
I have
on
one
what
which
you
is
and
sort
Nika
of
do?
relevant, and I wasn't going to raise it now, but I can because you've asked the question, so you've opened the door. But so you have a fantastic technology. It's really cool, your EPIC technology. And one of the issues that, of course, people have with the new technology is it does something no one else can do, so it's fantastic. But also, because no one else can do it, I've never tried it before, and I'm very weary. And science, which is, you'd think, the most innovative thing in the world, is also some of the times the most reluctant to new technology. So one thing that I would recommend is there a way new technologies can be tested? We're doing this right now with a particular company. Test them first. We were helping with beta testing. You've got a finished product. But can we test it until we publish with it? And then once we publish with it, then we give it back. And the next guy who wants to test it, then he publishes. Because as it becomes incorporated in the methodology, in publications, other people see when they go, oh wait, okay, I want to verify this, or I want to build on this. Oh, they're using this new technology at Corning, so I probably have to use that technology too if I want to verify it. Or they've got something that they're doing and they did it and I couldn't now. But if I had what they had in their methodology, I could. So that's like one gimmicky thing that I've sort of come up with on the fly to answer your question, but I hopefully that's one way to do it. So partnerships in that way, testing, publishing, and so we get a sneak peek and you get utilization, and eventually that sneak peek becomes a purchase somewhere.
Any more questions for Michael?
First off, great job, Mike. So a question for you about, you talked about being able to translate more of the research into actual development. So is that a matter of, do we train scientists to think more like businessmen? Do we teach them how to actually identify commercialization in their research? Or is it more about taking standing companies and pairing them together and saying talk? Is it more of a
well, sort of all, I mean, it's sort of all those. In one respect, is making sure that people which know things can be commercializable or can do it appropriately, so attorneys, are involved in the process. And so that's why having a strong procedures and protocols as an institution is important. For another thing, it could be making sure, like in competition settings, I know MIT does this, where they'll take a guy from Harvard Business School, they take an engineer from MIT, and they take somebody who was more particular to the basic science. And they plug them together. They make little teams. And your class is, you come up with something at the end of it. And so, like a lot of people, when your grade rides on something, or your notoriety rides on something, you tend to work you tend to work hard on it, but a product comes from that. But also, scientists become more familiar at an early age of having to work with businessmen, and businessmen become acquainted at an early age with how scientists work, because they're two people which oftentimes don't see the same thing the same way. And so the earlier you can introduce them as playmates, the better it becomes when they're actually in the major leagues. So I hope that answers everything today, and if we'd like, we can do lunch.
Thank you, Michael. Let's give him a round of applause and I'll. So we do have lunch available, and after lunch at 1 p.m. we're going to reconvene. Dan Carr is going to give the keynote, an interesting talk on kind of the interface of public-private science with respect to pain, which is arguably one of the world's, I think, is the United States' biggest problem right now in terms of health, social, and economic. So we look forward to that at 1 p.m., and lunch is out here. Please use the opportunity to network.